So this revision webinar is going to focus on the subject of cell biology, and we're going to be going through the topics in the AQA Advanced Level Biology specification, the current specification. However, what we're going to be going through is also applicable to those of you studying under OCR or EdXL or any of the other exam boards for advanced level biology. So just to quickly have a look at what we're going to be covering here. So this is the advanced level biology specification from AQA, the current specification. We're going to be going over the subject under the title topic 3.2 cells in this specification. I'll just make that a bit bigger. So we're going to have a rather detailed review of cell biology, starting with the structure of eukaryotic cells, the distinction between eukaryotic cells and prokarya, and we're also going to go into the structure of tissues, in other words, how these cells are organized into tissues in eukaryotic multicellular organisms. And we're going to then move on to look at the structures of prokaryotic cells and viruses. And we're also going to examine some of the methods that biologists use to study cells, including optical and electron microscopy, and also the processes of cell fractionation and ultracentrifugation. And then we'll move on to the next topic. Now, this topic here, within the cells topic, this subtopic 3.2.2, all cells arise from other cells, by prior arrangement with the private students at Invisible College, we're not going to be addressing this topic in this webinar, and the reason for that is that there is already a teaching video which you can access without enrolling on any course modules on my website, or you can access the same teaching video on the YouTube channel from my website. So if I bring up the YouTube channel here, I can show you where you can find that video, and that video will contain very detailed teaching on this topic here. So things like mitosis and the cell cycle will be covered in depth within that video. So let me just show you where you can find that. So here is the YouTube channel for my website here, and you can find the video either under the videos here. So if you go here, you will find a video on mitosis in the cell cycle here, which covers that subtopic. You're also going to find a rather detailed video on immunology, which covers the section of this topic in the AQA spec that deals with the immune system. Let me just see, where is that here? Here it is. So you can find this video here as well. So I'm not going to go over those two subtopics within this topic of cells, just because those videos have been on the YouTube channel for quite some time now, and you can find everything that you need in a lot of depth in those videos. And of course, if you have any questions, you can also ask your questions on the YouTube channel. Alternatively, if you go to my website, let me just bring that up here. So if you go to my webpage here, on the front page of the website, if you scroll down, there is a number of videos from the lessons of various course modules, which you can access for free without enrolling on any of those course modules, or without registering for an account on my website. So you can find, for example, down here, there's the video on immunology, and there's also the video on mitosis in the cell cycle. You can either watch those videos from here. So for example, you can click on this one and the video will play automatically, or you can scroll down further down the page, and you can find not just the video but also the actual lesson page, where you can find that video within its respective course module. So if you click down here, it will take you to the lesson page within the course module that I've created covering this entire topic on cells. Now, these two lessons within that course module are free and open to everyone, so by all means browse to this course module, which you can also reach by going to the course modules page and finding the course module on advanced level biology, cell biology. And if you click here, it will take you to the overview page, and you'll find that those two lessons are accessible free of charge and open for everyone. So again, there's one lesson on mitosis in the cell cycle. You can see the learning outcomes here. You can browse through some of the lesson slides here, and you can also click on this one and it will take you to the lesson which covers all of the ideas in immunology that you need to be aware of for this topic. So again, I'm not going to spend time on those topics in this webinar, and I've again, those of you who are private students and have booked tickets and are attending via the Zoom client, you're already aware of this. This is the topic on immunology. Again, all of the ideas here are covered in that video on immunology, so I encourage you guys, if you haven't already done so, to have a look at that. Now, I know that I believe all of you who are currently in attendance do have access to that course modules, and again, I've let you know of the kind of agenda for today in advance anyway. Now again, in keeping with the format of the previous webinars, if you do have any questions during this webinar, those of you who are in attendance, please do type those questions not into the chat box, but into the Q&A box instead on your Zoom client. The reason for that is that the chat box tends to scroll by very quickly, and it might be that if you type your questions in there, I may well miss the question. Whereas if you type them into the Q&A box, I will then look at the Q&A box at the end of the webinar when we turn off the YouTube live stream and we go into the Q&A section, and I will then address all of your questions in sufficient detail at the end of the webinar there, and I won't miss any of them out. Okay, so let's get started then. So we'll begin with a little bit of history. So the first thing that you all need to be aware of, and that hopefully you should have learned during the course of your GCSE biology, is that a cell is essentially the smallest functional unit of biology. In other words, it's the smallest piece of matter which fulfills all 7 characteristics necessary to define something as being a living thing. Now, cells, or the building blocks of life, were first discovered following the invention of the microscope, and this was one of the milestones of biology in the 16th and the 17th centuries. Now, it was actually Robert Hooke, which I've mentioned in various contexts, not just in biology but also in physics in previous webinars, who was responsible for, in large part, for these developments. And he was examining very thin sections of plant tissue under a microscope that he built himself. And he found that rather than being a continuous material, living materials were made up of small, what he described as compartments which resembled the dormitories that monks used to sleep in monasteries, which were called cellular. And so this is where we get the word cells from. And what you're seeing here is one of Robert Hooke's own drawings, which he made when examining a piece of cork under a microscope. And this is actually a photograph of the microscope that he himself designed and built and used to make these observations. So the cell is the fundamental living unit of biology, and as such, the modern 3-domain system of classification is highly dependent upon the structural properties and the features of the cells that make up different living organisms. So in general, these days, we use a combination of the physical features and also the genetic information belonging to an organism to classify which group that organism belongs to, and by extension which organisms it is most evolutionarily related to. And so we use this 3-domain system in modern biology. So let's have a look at these domains in sequence here. So we'll start with the domain of Eukarya. Now this is made up of all living things which are either classified as protists, fungi, lichens, plants, or animals. Now you can see here that before we divide all living organisms into kingdoms that many of you are already familiar with, before we do that, we first divide them into these differing domains. Now the domain of the Eukarya is the domain that contains these kingdoms. So, Eukarya generally will occupy a very diverse range of habitats, and they also have a very diverse range of modes of nutrition or feeding. Now, some members of this domain will obtain their nutrients by digestion and absorption, but others will be autotrophs. They will obtain their nutrients by photosynthesis, for example. Now, most members of this domain are also multicellular, although many of them are unicellular. So the example that's being shown here is the amoeba, which is a protist. So we also need to have a sense of the size of cells in general. So the question here is, how small is a blood cell? Now obviously cells generally are too small to be seen by the naked eye, so we use a microscope to look at them. And typically microscopes, when we're looking at cells, a useful unit of measurement for the sizes of these things is the micrometer, or the millionth of a meter. So 1 micrometer is 1,000th of a millimeter, or 1/1,000,000th of a meter. And so this diagram here on the left we can use this scale to compare the sizes of some structures to the sizes of some common cells. So let's start with the thickness of an average human hair, which runs at approximately 100 micrometers. Compared to that, one of the largest types of human cells, the ovum, the female gametes, is rather large. It's about the size of the thickness of a human hair. And then we have cheek cells, which are smaller, nerve cells, which, although they can be very, very long, they are rather narrow in terms of the size of their cell bodies or their axon diameters, for example. We also have sperm cells, which are smaller still. We have white blood cells and red blood cells. Now, red blood cells are on the order of 6 to 8 micrometers in diameter. So this shows that there's actually quite a range of sizes of cells found in the human body. Now, in comparison to Eukarya, prokaryotes are much smaller, typically. So if we take an average human red blood cell, we can see that the E. coli bacterium, relative to that, is far smaller in size. It's only about 2 micrometers in length, whereas the Streptococcus pneumoniae bacterium is even smaller than that. So typically a single coccus, and these are diplococci, which typically will pair up like this, but a single one of these is on the order of between 0.5 micron and 1.25 microns. So in terms of the arrangement of their genetic materials as well, eukaryotes are very different from prokaryotes. So the genetic material of Eukarya is usually restricted to a membrane-bound nucleus, and it's also folded and coiled like this around packaging proteins called histones into these packaging units called nucleosomes. Now, those nucleosomes are then further supercoiled and coiled further, and then structured into these structures called chromosomes for the purposes of cell division. So the DNA in eukaryotes is a linear molecule, and as you can see here in this diagram, it's wound around these nucleosomes. So each one of these nucleosomes will have around about 146 to 147 base pairs of DNA wound around it. Now, each one of these nucleosomes is made up of a complex of 8 packaging proteins, which are called histones. And this DNA and the octamer, altogether, the packaging proteins plus the DNA wound around them, is referred to as this unit called the nucleosome. Now, there are free ends to the linear DNA molecule which is found in eukaryotes, but these ends are called telomeres and have structures on them called telomeres. Now, cells that contain these free ends have developed rather elaborate methods of DNA replication, including mitosis and meiosis, which you can go into in the previous webinar on genetics, diversity and inheritance, where we talked about meiosis, but you can also have a look at the actual mechanisms by which these chromosomes are manipulated during mitosis in that video that I showed you before as well. Now, bacteria typically are much simpler than this. They do not have these very complicated mechanisms for the replication and the distribution of their genetic information. Oh, incidentally, if you're hearing a little bit of background noise, again, the reason for that, unfortunately, is that it's very, very hot in London at the moment, so I'm having to open a couple of the windows, and so you may hear a little bit of background noise in the background. Those of you who are attending via the Zoom application, if that does become too distracting for you, please do let me know, and I will see if I can do something about that by changing either the mic gain or maybe closing the windows and doing something else. So please do let me know if that becomes too distracting for you. So, speaking of genetic information, the organelles such as mitochondria and chloroplasts actually do contain their own loops of DNA, their own genetic information, and they are able to reproduce independently of the cell. So, the mitochondrial DNA, it actually resembles the circular genome that you find in bacteria. And so, within the mitochondrial matrix here, you actually find these circular loops of DNA, and these circular loops of DNA allow the mitochondrion to replicate itself independently of the cell that you find it in. So they also have some other features which also resemble prokaryotes. So they also have small ribosomes, for example. Now, all of these different features that make them resemble prokaryotes have led to what's called the endosymbiont hypothesis. Now, this is the hypothesis that early in evolutionary history, these were once free-living prokaryotic organisms which became engulfed by eukaryotic organisms, and instead of being broken down by them and digested, they became symbiotic with them, so that the eukaryotic organism provided them with shelter from the surrounding conditions, and in turn the prokaryotic organisms manufactured large amounts of ATP for use as an energy source by the eukaryote. So the endosymbiont hypothesis is that organelles such as mitochondria and also chloroplasts, which share some of these features, were once free-living prokaryotic organisms early in evolutionary history, and became engulfed by the early prokarya, or the ancestors of the prokarya, of the eukarya rather, and struck a symbiotic relationship up with them. So, in addition to the chromosomes, there's also short lengths of DNA within the chloroplasts as well. So again, we see here that chloroplasts also have their own ribosomes and their own circular loops of DNA. So again, this makes them resemble somewhat bacteria and prokaryotic organisms. And again, this has led to the idea that these also were once free-living organisms, which struck up a symbiotic relationship with the ancestors of the modern eukaryotic organisms. So speaking of ribosomes, these are the organelles where protein synthesis takes place, the process of translation. Now, eukaryotic ribosomes are in general larger than prokaryotic ribosomes. Both types of ribosomes are made up of two subunits, a small subunit and a large subunit. In the case of the eukaryotic ribosomes, they have a larger size. Now, the size of ribosomes is given in units of Svedberg units, or S, as you can see here. So eukaryotic ribosomes are 80S in size, or 80 Svedberg units in size, whereas prokaryotic ribosomes are typically much smaller. They're around about 70S. Now, it's important to note here that the Svedberg unit is not a kind of linearly additive unit like length in meters or something like that. It's a unit which measures how quickly these particles sediment or undergo sedimentation when they're in a suspension. And so you can see here that the small subunit of a eukaryotic ribosome is of a size of 40 Svedberg units, the larger subunit is of a size of 60 Svedberg units, but these two don't add together to make 80S. And so the Svedberg unit is just a unit which is used to describe the size in terms of how rapidly these particles sediment or undergo sedimentation when they're in a solution or a suspension. So, as we can see here, by direct comparison, the prokaryotic ribosomes are typically smaller. So let's summarize the features that we've been talking about here. Eukaryotic cells tend to be larger than prokaryotes. They tend to also have cytoplasms which have membrane-bound organelles. Now, this is actually the biggest single feature which distinguishes eukarya from prokarya. So typically, prokaryotic organisms do not have internal membrane-bound organelles. They do sometimes have infoldings of the plasma membrane within which certain biochemical and respiratory reactions occur, but those aren't really internal organelles. So for example, in a prokaryotic organism, as we'll see later on, the genetic material floats freely in a region of the cytoplasm in the center of the cell and is not surrounded by a nuclear envelope. Prokaryotic organisms do not contain other membrane-bound organelles such as Golgi apparatus and smooth and rough endoplasmic reticuli, for example. They simply have no internal organelles that have membranes around them. Also, as we saw, the ribosomes in eukarya are larger than those found in prokaryotic cells. And of course, a eukaryotic cell has a true membrane-bound nucleus which contains linear DNA. And this is in contrast to the prokaryotic DNA, which exists as a continuous loop. Also, the cell wall, which is found in some eukaryotic cells, those of plants for example, is made up of a very different material than the material which makes up the cell walls of prokaryotic organisms, which is made up of a peptidoglycan. Also, fungi, which are eukaryotic, also have a very different material making up their cell walls than you find in prokarya as well. So, we mentioned that eukaryotes are distinguished, we distinguish between eukarya and prokarya in general by the absence of membrane-bound organelles in prokaryotes. So we can see some of these membrane-bound organelles in this diagram here. So we have the nucleus, the mitochondria, the Golgi apparatus, the secretory vesicles, which come off the Golgi apparatus and also off the rough and the smooth endoplasmic reticuli, the rough endoplasmic reticulum and the smooth endoplasmic reticulum as well. So if we're looking at a generalized eukaryotic cell, there are certain features that we find in all eukaryotic cells. We find a plasma membrane, we find the cytosol, we find suspended within the cytosol various types of membrane-bound organelles that we mentioned before, but also remember there is a distinction between the term cytosol and the term cytoplasm. The term cytosol refers to the kind of jelly-like watery matrix that makes up the liquid component of the internal portion of the cell. Now the cytosol plus the internal organelles, this is what we refer to as the cytoplasm. So basically everything within the bounds of the plasma membrane is referred to as the cytoplasm, but the watery kind of gel-like liquid that you find within that, in which the organelles are suspended, that is called the cytosol. Now the plasma membrane is a very important organelle. It is the organelle which controls the movement of substances in and out of the cell, and also is responsible for detecting changes in the cell's external environment as well. Now we're going to examine the plasma membrane in a little bit more detail later on in this webinar when we start to talk about the transport of substances across that membrane. So if we're looking at the plasma membrane, we can magnify at approximately 250,000 times, and then the structure of the plasma membrane becomes a little bit more visible under electron microscopy, for example, and we find that it's made of a lipid bilayer. It's made up of a layer of two layers of phospholipid molecules, which are oriented so that their hydrophobic tails face each other in the internal portion of the membrane, and the thickness of the membrane is approximately 7.5 nanometers. Now within this membrane, as we'll see later on, we have various protein molecules and other modified proteins and other substances such as sterols like cholesterol embedded within the membrane and also on the outer and inner surfaces of the membrane. So if we're looking at the nucleus, all eukaryotic cells possess in general a nucleus, although there are a few exceptions such as mammalian red blood cells and things like that. And the nucleus is a rather large organelle, and it's characterized by having a double membrane, a double envelope, which has pores in it. Now these pores will open and close to allow messenger RNA to leave the nucleus and enter the cytoplasm, where it can undergo translation by ribosomes. You can also see here that the external surface of the nuclear envelope is actually continuous with the rough endoplasmic reticulum, and like the rough endoplasmic reticulum, you also find ribosomes dotted along the outer surface of the nuclear envelope. Now, it's also important to remember that chromosomes, although they are important structures, these are not visible through the majority of the cell cycle. These only become visible during the period leading up to cell division and during cell division. For the majority of the rest of the time, the genetic material in the nucleus is packaged into a material called chromatin, and chromatin is a dense, dark staining material which you can kind of see as blobs in the nucleus during the portions of the cell cycle where the genetic material is not undergoing replication, or during the portions of the cell cycle where the genetic material is not being moved about during cell division. And remember, the chromatin is made up of the DNA plus those packaging proteins as well. So here we can see the nuclear membrane. It's a double envelope and it has these pores. The the fluid inside is called the nucleoplasm, and the densest region, the densest staining region within the nucleus is called the nucleolus, and this is where the majority of the chromatin can be found to be packaged. So this is a diagram showing the nuclear envelope in a little bit more detail. Now, the distance between these two adjacent membranes is approximately 40 to 70 nanometers, and between these membranes you have a space called the perinuclear space. And the pores, as we said before, these enable messenger RNA to move out of the nucleus and into the cytoplasm, for example. And there's also other forms of communication that occur between the nucleus and the cytoplasm via these nuclear pores as well. And also you can see that there are ribosomes which are embedded within the outer nuclear membrane. And again, the nuclear membrane itself is continuous with many portions of the rough endoplasmic reticulum. So here we can see an electron micrograph, which is a transmission electron micrograph showing the nucleus. We can see the dense staining chromatin material and the large collection of that material in the nucleus that we call the nucleolus. Now, the nucleolus is a dense, spherical, heavily dark staining region of the nucleus, and it's not a membrane-bound structure. Now, you find that it's the ribosomal RNA, or rRNA, which undergoes transcription and processing in the nucleolus, and it's actually here in the nucleolus where the ribosomes are assembled. So again, the genetic material of all eukaryotic cells consists mostly of long linear DNA molecules which are packaged into these units called nucleosomes by winding them around these octamers of histone proteins. These are then packaged further by coiling and supercoiling, eventually to form these structures called chromosomes. And again, the free ends of the chromosomes are referred to as telomeres. So, the mitochondria are responsible for producing ATP, a chemical which is sometimes referred to as the energy currency of the cell. Now, the ATP, the adenosine triphosphate, is the molecule which is used within the cell to power cellular processes. So it's a source of energy for the processes that occur within the cell that require energy. So sometimes you might see mitochondria loosely referred to as the powerhouses of the cell. So let's look at these in a little bit more detail now. So a mitochondrion does actually also have a double membrane. You have an outer membrane and you have an inner membrane which is folded into these protrusions which increase the total surface area of the inner membrane. These are called cristae, these foldings. And the inner fluid within that inner compartment is referred to as the mitochondrial matrix. Now, the inner surface, the inner membrane, is where many of the very important aerobic biochemical reactions that are involved in respiration take place. And so typically you will find many proteins embedded within the inner mitochondrial membrane, but you will also find, I'll just go back here, that when you study the inner mitochondrial membrane under an electron microscope, you can see that it's studded with these minute spheres in this manner. So here we can see the mitochondrion at under a transmission electron microscope. We can see clearly here the infoldings, the cristae of the inner mitochondrial membrane. We can also see the fluid matrix which fills that inner space. And we can also see the outer membrane, and outside of the mitochondrion here we can see a portion of the endoplasmic reticulum within this cell as well. So, the rough endoplasmic reticulum and the smooth endoplasmic reticulum, the word reticulum means essentially, it's equivalent to the word network, and endoplasmic means within the cell, essentially. So, this is a system of flattened sacs which extend throughout the cytoplasm. Now, the rough endoplasmic reticulum is studded with ribosomes, and again, the ribosomes are the organelles where protein synthesis occurs, and so many of those proteins are stored within the rough endoplasmic reticulum, where they can then be transported to various portions of the cell to be used or be modified further. So this is a diagram showing that endoplasmic reticulum and the membranes that make it up in more detail. Between those membranes you have the space called the cavity of the endoplasmic reticulum, and the flattened membranes themselves are referred to as lamellae. This is an electron micrograph showing the same thing here. So the smooth endoplasmic reticulum is similar, but the difference here is that it contains no ribosomes embedded within its membrane. Now, it does also have a slightly different morphology. It looks a bit more like this than a system of flattened sacs or flattened lamellae. Now, the function of the smooth endoplasmic reticulum is for the synthesis of lipids and steroids, and also fatty acids and things like that, and also their transport around the cell. Now, the Golgi apparatus is an organelle which is made up of between 4 and 6 of these flattened sacs. And so you see here the Golgi apparatus here, somewhere between 4 and 8, of these membrane-bound sacs. And usually you find the Golgi apparatus somewhere near the nucleus of the cell. Now, typically, the function of the Golgi apparatus is the processing and the modification of proteins and also lipids which are being produced within organelles such as the rough endoplasmic reticulum and the smooth endoplasmic reticulum. And the processing of those substances happens within these sacs of the Golgi body, and eventually, once the processing of those substances has been completed, small secretory vesicles will pinch off the ends of these sacs and then be transported to either the membrane, where they can be exocytosed, or to other parts of the cell. So many of the secretory functions of cells, in other words, many of the substances which are produced and are destined to be secreted out of the cell into the extracellular space, are produced and modified within the Golgi apparatus. Now, these flattened sacs of the Golgi apparatus, they act as a kind of assembly line, where let's say you have a protein from the rough endoplasmic reticulum which needs to be modified in order to be prepared for its proper function. Well, a vesicle will pinch off the rough endoplasmic reticulum containing that protein. It will be transported and will fuse with what's called the cis face of the Golgi apparatus, and each of these flattened sacs is referred to as a cisternum, the plural being cisternae, and it acts as an assembly line where there's a different series of biochemical modifications that take place using different enzymes in each of these cisternae. So once the first phase of modification has been completed in this cis face cisternum, a vesicle will pinch off here containing the modified protein or lipid, and then will fuse with the next face. Another set of biochemical modifications will take place inside this cisternum, at which point the protein is then transported to the next one via a vesicle pinching off here, and so on until it reaches the trans face of the Golgi body. And at this point, the modification of that protein or lipid has been completed. The vesicle, secretory vesicle, can then pinch off here and move towards the membrane for secretion, or to some other part of the cell for that chemical to perform its function. So here is another diagram of the Golgi apparatus, and here we can see the membrane-bound cisternae stacked on top of each other. And typically you will find lots and lots of these secretory vesicles floating around in close proximity to the Golgi apparatus. So we can see here that we have like these satellites of vesicles surrounding this. Now, typically in multicellular organisms where you have cells within tissues which are responsible for the secretion of substances, such as, for example, the cells found in the walls of the digestive system, you will find cells which have very highly developed Golgi apparatus. This is a transmission electron micrograph showing the Golgi body, as you can see here. And here you can see the satellite of vesicles surrounding the Golgi body. So, we've been talking about the general features of eukaryotic cells, in other words, the features that all eukaryotic cells possess. Let's now take a look at the features which are unique to animal cells. So, there are, at the cellular level, there are key differences between the kingdoms of the domain Eukarya. So, there are key differences between plant and animal cells, and differences between plant and animal and fungal cells, and so on. So we're going to look at some of those differences. So for example, plant cells have a cell wall. They have a large central vacuole containing a fluid known as cell sap. They also contain chloroplasts, whereas these structures are not found in animal cells. Also, conversely, animal cells also possess a number of features which are not found in plant cells. So, for example, many animal cells possess these finger-like projections, these foldings of their plasma membranes, which are referred to as microvilli. They also have organelles called centrioles. Now, centrioles are involved in the formation of the spindle and the spindle fibers during cell division. And again, you do not find these in plant cells. So, plant cells have cellulose cell walls. They have large vacuoles that store cell sap, and they have chloroplasts, whereas animal cells possess sometimes microvilli. They have centrioles. And these are unique features to each of these two kingdoms. So the microvilli are foldings of the plasma membrane which you find on some animal cells, particularly, for example, on these cells which line the cavities of the body where absorption is taking place, like, for example, the small intestine. And the microvilli, the function of these is to increase the surface area available for the exchange of substances across the plasma membrane. So again, here we see the plasma membrane, here we see the microvilli and the cytosol, the nucleus, and some of the other organelles that we've already referred to. Now, another aspect of the internal ultrastructure of eukaryotic cells in general is the possession of cytoskeleton. Now, this is not something which explicitly appears in the advanced level biology specification for AQA, but it is something that's quite interesting to know about. Now, you might have wondered in the past, what is it that causes, for example, secretory vesicles to move towards the membrane for secretion? What can cause that if all that you find inside here is just a fluid gel? Well, in fact, it's a little bit more complex than just a simple fluid. Throughout the cytoplasm, you have these tiny filaments made up of proteins, and there are various types of these. There are microfilaments, there are intermediate filaments, and there are larger filaments called microtubules. Now, these are all made of different kinds of protein, and some of them are contractile. They can actually contract and shorten and then lengthen. Now, the function of these is for the internal cellular motion. So, for example, if you want to move, or if the cell needs to move one of these secretory vesicles towards the membrane, it's the movements of the cytoskeleton that enable that to happen. But also, if you've ever seen a video or looked at an amoeba under a microscope, you'll notice that it's able to change the shape of its membrane to engulf particles like smaller unicellular organisms. And the way that it can do this, and the way that it can do this endocytosis and pinocytosis, is again by means of the cytoskeleton. Now, the cytoskeleton is something that we're not going to go into in depth, or at all, in fact, beyond what I've just said in the course of this webinar, just because, again, we're sticking to what's present in the advanced level specification for AQA. But if you are interested in more detail on that, again, I would encourage you to go to this course module on cell biology on my website, where in the first few lessons we examine many of these organelles in a lot more depth. So again, I know those of you who are in attendance in the private Zoom room, many of you already have access to this, and the LSWAP consortium students already do. But in the first few lessons, particularly the one on animal and cell structure, we go into the cytoplasm in a lot, the cytoskeleton rather, in a lot more depth there. So if you are interested, please do check out that course module as well. So there are also membrane-bound vesicles known as lysosomes, which contain acid hydrolase enzymes. Now, these are the function of these is for internal or external cellular digestion. So, for example, these will fuse with vesicles containing bacteria such as phagosomes, which are produced by phagocytosis, and the contents of the lysosome containing acid hydrolases such as lysozyme will mix with the contents of the phagosome, and the enzyme within here will then digest and break down that bacterium, and then the products of that digestion can be absorbed and used by the cell, and any waste products can then be exocytosed. And also, when cells become damaged or tissues become damaged by disease, for example, or injury, these lysosomes will then break open and start to break down that cell's internal contents. So, for example, we have bacterial cells here being engulfed when the plasma membrane invaginates itself to form a phagosome, like this. And so this is a phagocytic vacuole. And then we get a primary lysosome which then fuses with this vacuole like this. The contents will then mix together to form a secondary lysosome. And in this secondary lysosome, the bacterium or whatever it is that's just been ingested by or endocytosed by the cell is broken down by the hydrolytic enzymes found in the lysosome. Then some of these useful products of that digestion are absorbed into the cytosol and are used by the cell, but some of them can then be exocytosed and moved out of the cell. So this is an example of lysosomes being used for intracellular digestion taking place inside the cell. You can also destroy aging organelles by this same process. So another function of these lysosomes is to break down organelles which have reached the end of their useful lives and need to be broken down, and their constituent parts, the proteins and so on, recycled to form new organelles. So another function of lysosomes concerns cytoplasmic turnover. So when a cell organelle ages or becomes redundant, it becomes surrounded by a membrane to form a vacuole, like this. A primary lysosome will then fuse with this to form a secondary lysosome, where the contents of the lysosome and this vacuole will then mix. The redundant organelle is then digested by the enzymes from the lysosome to form a secondary lysosome, and the products of that digestion are then absorbed by the cell, and some of the waste products which are not needed by the cell form a residual body, which is then exocytosed to eject those waste products out of the cell. So another function of lysosomes is that the digestive enzymes found in the lysosomes can be used for extracellular digestion. So they can simply be moved towards the plasma membrane, they fuse with the plasma membrane to exocytose the contents of the lysosome into the extracellular space, where the enzymes can then digest particles or other cells in the extracellular space. So, for example, certain types of bone cells or osteoclasts will release enzymes to destroy cartilage tissue, which can then be replaced by bones, and this type of activity occurs during development. Now, when the entire cell becomes diseased or damaged, the entire cell can be destroyed by the action of lysosomes as well. So, once this happens, once a cell becomes diseased or damaged, the response is that the lysosomes themselves, the membranes of these lysosomes, become leaky and permeable, and then the contents of the lysosomes will then leak outwards into the cytosol, where they start to break down all of the internal organelles of the cell and essentially kill that cell and break it down. So this function in bringing about programmed cell death or apoptosis, this is called autolysis. Autolysis meaning self-splitting or self-breaking. So lysosomes are sometimes loosely termed the suicide bags of the cell for this reason. So we're coming up to about 50 minutes into the first hour of this webinar, so or rather about 55 minutes at this point. So I'm going to give you guys a 5-minute break, and then after the break, when we come back, we'll get into the specifics of the special features of plant cells. So I'm going to put the timer up on the board shortly, and I will see you in about 5 minutes time. Okay, so let's go into the special features of plant cells now. So as we said before, plant cells are characterized by having a cell wall which is made up of a structural carbohydrate called cellulose. So not all eukaryotic cells have cell walls. Now typically it's the plant and the fungi which have cell walls. Plant cell walls are made up of cellulose, whereas the fungi have cell walls which are made up of a different material called chitin, or chitin, depending on who you ask. So, as in all eukaryotes, plant cells possess a plasma membrane, and within the plasma membrane there is a fluid called the cytosol, within which all of the membrane-bound organelles are suspended. But unlike animal cells, they do also have a cell wall around that cell membrane. So here we have the plasma membrane, this thin line here, and the thicker blue line in this highlighted cell here is the cell wall, and within we have the cytoplasm. Now these are slightly off, these labels, I'm not sure why, apologies for that. So typically between the cell walls of adjacent plant cells, sometimes you will have these channels where the cytoplasm or the cytosol of one cell communicates directly with the cytosol of the adjacent cell. These are called Plasmodesmata, but also between adjacent cell walls, these cell walls in plant tissues are typically glued together by a substance called pectin. So, the cell walls of plants are made up of cellulose, and the cellulose lays itself down as fibres, which will overlay each other to form this fibrous cell wall structure here. Now, in these revision webinar on the subject of biological molecules, we went into the subject of the structure of cellulose in a lot more detail, but the matrix of the cell wall also consists of materials called pectins, such as calcium pectate, but also other materials called hemicelluloses. So when a young plant cell is formed, it's surrounded by a primary cell wall, which is made up of largely just cellulose, and the cell wall is actually secreted by the protoplast which it encloses. So the primary cell wall begins by being composed of cellulose fibers arranged in a random manner. And this is actually a micrograph of these randomly arranged cellulose fibers. Now this initially, during the young phases of the cell's development, this randomized coiled configuration of the cellulose fibers allows the cell wall to expand as the young cell becomes larger and grows. So as the plant cell grows, more and more cellulose fibers are secreted by the protoplast and are laid down on the inside of the primary cell wall, and this forms a secondary cell wall. Now the cellulose fibers of the secondary cell wall are arranged in a much more geometric fashion, in a much more patterned manner, and this allows them to give considerable strength and rigidity to the cell wall. So this diagram here shows the kind of arrangement in the secondary cell wall. As you can see, it's much more ordered. And this electron micrograph shows what these cellulose fibers in the secondary cell wall actually look like. Now it's important to remember that unlike the cell membrane, the cell wall is porous and it's freely permeable to all of the substances which the cell needs to transport. So these would include water, ions, sugars and other essential nutrients, but also waste products which the cell needs to get rid of by passing those products into the extracellular environment. So the cell wall is freely permeable, whereas the cell membrane is selectively permeable. So in many cases where adjacent plant cells are held together in a tissue, there is communication between the cytosol of adjacent cells. So we can see here that the adjacent cells in this diagram are interconnected by these fine strands of cytoplasm, and these are called plasmodesmata. Now these can pass through the pores in the cell walls and also the middle lamellae between those cell walls. And so essentially these form these very thin cytoplasmic bridges between one cell and another. So the function of the cell wall in general is to provide mechanical support and also protection for the plant cells. So if we were to look at this region of these two adjacent cells under an electron microscope, we might see something like this. So here we can clearly see the Plasmodesmata, which communicate between the cytosols of the adjacent plant cells through their cell walls and through this middle lamellum here. Now, for some reason, all of these labels have gone a little bit out of whack, so apologies for that. Now, another one of these features that all of these plant cells share in common is a very large central membrane-bound space containing cell sap, a fluid called cell sap, and this is called the large central vacuole. So, this is a fluid-filled membrane-bound sac, and cell sap is a concentrated solution of many different dissolved substances in water. Now, the membrane that surrounds the central vacuole is called the tonoplast, and the vacuole functions as a storage region for the cell, but also is responsible for maintaining the shape and the volume of the cell. It's also responsible for providing rigidity to the cell, because when there's sufficient fluid in the cell, this produces a pressure which causes this cell, the highlighted one in the middle here, to push on the surrounding cells with its cell wall, and similarly the surrounding cells do this also. So all of the cells within the plant cell's tissues will push against each other, and that collectively provides the rigidity in parts of the plant, like the stem of the plant, to provide support for the plant so that it doesn't wilt. Now, if the plant loses too much water, typically the large central vacuole will shrink, and that outward pressure is diminished, and this is what leads to the plant losing its rigidity and wilting. So many plant cells also possess organs called amyloplasts. And amyloplasts, like these are organelles which convert glucose into another substance called amylopectin, and amylopectin eventually forms the storage carbohydrate starch. Now, also many plant cells will contain chloroplasts, which are the site of photosynthesis. So let's have a look at chloroplasts in a bit more detail. So these have a double membrane envelope, and the jelly-like matrix within the chloroplast is referred to as the stroma. Now, within the chloroplast, you also have stacks of membrane-bound vesicles called thylakoids, which are stacked into these grana and have these connecting lamellae between them. So here we can see the thylakoids. Now, each of these is a membrane-bound sac containing various photosynthetic pigments, such as chlorophylls. Now, these are stacked in these arrangements called grana. The singular is granum, so this would be one granum here, this would be another granum here, and between these you have these thin extensions called lamellae. So this would be a single granum. Now, as we said before, chloroplasts are similar to mitochondria in the sense that they also contain these circular loops of DNA, which allow these organelles to replicate themselves independently of the cells that you find them in. They also contain their own ribosomes. You also find lipid droplets inside the stroma of the chloroplast as well. Now, many of the sugar molecules which are formed during photosynthesis can be stored as starch, and you often find these very large starch grains within the chloroplasts, and this is a good way of storing large numbers of glucose molecules which have been produced by photosynthesis. So this is a photomicrograph showing the detail of the internal contents of a chloroplast, and you can clearly see the grana with their stacked thylakoids and their lamellae. You can also see the starch grains as well, and you can also see the tiny little ribosomes which are dotted around within the stroma. So we mentioned that the thylakoids, these membrane-bound sacs, contain many of the photosynthetic pigments such as chlorophylls. So during photosynthesis, the chlorophyll molecules will absorb photons, packets of light energy, and the electrons in those chlorophyll molecules are then excited to higher energy levels, and it's this process which feeds energy into the process of photosynthesis and down that biochemical sequence of reactions which leads to the production of ATP. Now again, if you want more detail on that, you can have a look at the webinar that we did earlier in this series on the subject of energy in biological systems. Now the reason why you get these thylakoids stacked into these arrangements called grana is for the maximum absorption of sunlight energy, because the light energy, instead of just passing through thylakoids which are randomly arranged, it has to pass through successive thylakoids arranged in stacks, and this ensures that the chloroplast absorbs the maximum amount of light energy for photosynthesis. So the reactions that require the light energy are referred to as the light-dependent reactions, and these involve the conversion of the absorbed light energy into chemical potential energy, and that chemical energy is then used by the chloroplast eventually to make organic food substances such as glucose, but also the glucose can be then assembled and modified into other essential nutrients such as amino acids and fatty acids. Now the series of chemical reactions leading to the synthesis of the food actually takes place in the stroma, the fluid component within the chloroplast, within which all of these thylakoids and grana and other components are suspended. So these reactions are referred to as the light-independent reactions because they take place and do not require the presence of light. So we've looked at some of the special features of plant cells. Let's now have a look at some of the ways in which animal and plant cells are organized into tissues. Now again, I'm not going to go into this in quite as much detail as I do in the course module on cell biology on my website, just to save time, and also because all of you in attendance who've booked tickets also have access to that course module. Now you'll also find a lot more detail on these in the revision notes and the other learning and study resources which you've received having booked tickets for this webinar as well. Many of you have already uploaded those to your private coaching areas on my website in that course module, and you've already received feedback and marking on those. Those of you who haven't, I would encourage you to do so, because again, it's one thing to do those exercises and to have the mark schemes for the exam questions and everything, but it's also useful to have some feedback from me and also some marking from me as well. So if you haven't already uploaded your work and the answers to some of the exercises in your revision and study resource packs which you received for booking a ticket for this webinar, then I'd encourage you to go into your private coaching area on my website and upload it there, and typically I will get back to you with marking and feedback within 24 to 48 hours. So let's have a look at cellular organization in general in multicellular organisms. Now, in biology, we talk about the ways in which biological structures are organized in a series of nested hierarchies. Now, biological systems are the most complex systems which scientists have encountered, regardless of what science you're talking about. So whether we're talking about chemistry, physics or biology, of all of these sciences, typically the biological systems are by far and away the most complex and the most diverse systems for us to understand. Now, because they're so diverse and because they're so complex, we need to start to break them down and be able to talk about them meaningfully in terms of categories and hierarchies. And so we start with the cell as the fundamental living unit of biology, but cells in multicellular organisms are organized into tissues, and those tissues are organized into organs, organs into organ systems into multicellular organisms. But we can even go beyond that, because in the science of ecology, we talk about organisms being organized into populations, populations into communities, communities into ecosystems, and so on and so on and so on. So we start with the fundamental living unit of biology, the cell. Now cells can differentiate and specialize into specialized cells which perform specific functions and have specific morphologies, specific shapes and structures. But then specialized cells of a similar type can group together to form a tissue which carries out one or more specific functions. But then you can have different tissues which are organized into discrete structures called organs, which carry out specific functions within a multicellular organism. So you can then organise different tissues and organs together to form an organ system which carries out one or more definite functions within the organism as a whole. But then all of the different tissues, organs and organ systems work together to form an organism which carries out all of the characteristics of living things. So these, if you like, are the levels of organisation and the definitions of some of those levels. So in larger multicellular organisms, many cells will specialise to carry out a particular function. Now these specialised or differentiated cells will group together to form tissues and organs. So we can define a tissue as a group of similar cells which associate together and are specialised for a particular function. Now I'm going to pause here and ask the question to those of you in attendance in the private Zoom webinar room. How would you define the words specialisation and differentiation in biology? Now this is something that we covered in a previous webinar in this series, but I think it's worthwhile mentioning here because again this is one of those things that students typically will drop marks on because they use these terms either incorrectly or define them incorrectly, or they use them interchangeably when they are in fact they do in fact have very different, very rigorous definitions. So how would you define those two terms? This will give me a chance to have a sip of my tea because once again I'm losing my voice. Okay, so we have a couple of responses here, seem to be on the right track. So a couple of you have defined what differentiation is. So how would you define specialization? So we have one suggestion here that differentiation is when a stem cell changes to become a cell of a particular type of tissue. Specialization is the adaptations of a cell. Okay, so you guys are on the right track. Now, obviously, there are many different ways you can kind of structure your definitions of these things, and they can all be correct as long as they mention the correct things. So, in general, just remember that differentiation is the process by which a cell becomes specialized. So, for example, if you have an undifferentiated or unspecialized stem cell, for example, in the bone marrow or in the growing embryo, that cell does not have any of the specific features of any specific tissue. However, when it differentiates, it undergoes a series of changes which switch on certain genes within the genome of that cell and switch off certain other genes so that the cell only expresses the characteristics which enable it to perform the functions of a particular tissue. So, for example, a hemopoietic cell in the bone marrow, one of the stem cells that you find there, can undergo differentiation in order to become a specialized red blood cell or white blood cell, for example. So the point that I'm making here is that differentiation is the process by which a stem cell, for example, will change to become specialized to perform a particular set of functions. So differentiation is the process by which specialization occurs. Specialization refers to the specific structural characteristics of a cell which enable it to perform specific functions within a multicellular organism. So a single red blood cell we see here, but if you have many red blood cells together, so for example like this in blood, you have a tissue. So blood, even though it's a liquid, is in fact a tissue according to the definitions that we've given for these terms. So a tissue, again, is a group of specialized cells which are specialized to perform specific functions within an organism. So tissues such as muscular tissue and nervous tissue, these can join together to make an organ such as a heart. So the heart is made up of a complicated arrangement of various kinds of tissue. You have cardiac muscle tissue, you have connective tissue, you have various types of specialized nervous tissue within the heart, but all of these are collected together and structured in such a way that they form a discrete functional unit within the organism that performs a specific function, in this case the pumping of blood to and from the lungs and also to and from the rest of the body. So, a system or organ system is made up of several organs and tissues that work together within that multicellular organism to perform a specific set of functions which are not performed by other organs or other tissues. So, for example, the stomach, together with other organs such as the liver and the small intestine, for example, these form what we refer to as the digestive system, whereas the heart, together with other organs and tissues such as blood vessels, these form the circulatory system. So an organism, such as ourselves, contains all of these organ systems working together. So, we mentioned that a tissue is a group of specialized similar cells which work together to perform a particular function or functions. Now, in animals, there are 6 principal kinds of animal tissue, which you should be familiar with to some extent. So, there is epithelial tissue, there is connective tissue, there's skeletal tissue, there's blood tissue, there's muscle tissue, and there's nervous tissue. So all of these different kinds of tissues, many of these can be found in the same organs. So the stomach, for example, is an example of such an organ. It contains several tissue types. So you'll find, for example, in the walls of the stomach, you'll find muscular tissue. You'll also find blood tissue, because the stomach contains blood vessels. You'll also find nervous tissue. You'll also find connective tissue. You'll also find epithelial tissue lining the stomach as well. So the stomach, like all organs, is a complex set of different types of tissue which are organized in a certain anatomical arrangement which allows that organ to fulfill its functions within its respective organ system. So let's take a look at epithelial tissue. Now, epithelium is the simplest kind of tissue that you find in animals, and there are several variants of this, and each of these types of epithelium perform different specific functions. So all epithelial tissues are tissues that line or surround the edges of organs, and this includes the external organ of the skin. Now, epithelial tissue forms an inner lining also of many tubes, ducts, and internal cavities and spaces. Now, in that context, the epithelial tissue is referred to as endothelium, endo meaning within. So epithelial tissue is made up of a sheet of cells which are found on a basement membrane. Now, the cells will fit closely together like the slabs in a pavement that you see here. Now, in fact, one kind of epithelium is actually called pavement epithelium because it has much the same organization as you see here on this photograph of a pavement. So, simple epithelium consists of a single layer of cells, and there are several distinct types of epithelium. Now, complex epithelium, in contrast to this, consists of multiple layers of cells. Now, this arrangement of multiple layers of cells, biologists will describe this as stratified epithelium. So, for example, in geology, stratigraphy is the study of the layers of geological materials like rocks, and we use the same word in a similar way in biology. So here we can see simple cuboidal epithelium. This is a diagram showing squamous or pavement epithelium, where we still have a simple single layer of cells resting on a basement membrane, but these are thinner and flatter and are arranged in that kind of crazy paving manner that you saw in that previous photograph of that pavement in that previous slide. This is columnar epithelium, and it's called columnar epithelium for obvious reasons. Here we can see stratified or complex epithelium, and this is the kind of epithelium that you find, for example, in the surface of the skin. Here we can see glandular epithelium, which is columnar epithelium where some of the cells have a secretory activity, and this is the type of epithelium that you typically find, for example, in the inner lining of organs like the stomach. And you also have ciliated columnar epithelium. So this is columnar epithelium which has this brush border, and again you can find this in lining various internal tubes and cavities, such as, for example, the airways within the respiratory system. Now, we go into many examples of where you can find these in that course module on cell biology, so those of you in attendance who want a bit more detail on this and some more examples, please do look in there, but in the interest of time I've just summarized them diagrammatically in this manner in this webinar here. So let's have a look at some of the tissues very briefly here. Now, connective tissue in contrast to epithelial tissue, this is the type of tissue which, funnily enough, connects other tissues together. So this types of connective tissue are things like ligaments, tendons, meninges, which are the connective tissue membranes that you find surrounding the brain inside the skull. You also find connective tissue in the context of collagen, bone marrow, fat cells, and various other examples as well. So for example, and vegetarians and vegans, you might want to turn away at this next diagram here, but for example, if you take a slice of beef or a steak, you can see that the connective tissue is these white sections that you see here, whereas the muscular tissue are these reddish sections that you see underneath. So skeletal tissue supports and protects the body's delicate organs. So for example, in the thorax you have the rib cage, which protects the organs of the chest cavity, but you also find the skeletal tissue which in the context of allowing movement. So you find skeletal tissue inside the limbs as well, where the muscles of the limbs will attach and allow movement of those limbs. So the function of the skeletal tissue in the skeletal system is both support and protection and movement as well. So here we can see a section of bone tissue at 1,000 times magnification through an optical microscope, and here we can see the bone cells, or the osteocytes. Now the osteocytes will secrete a mineral substance called calcium phosphate, and you can see that here. And this organization is what gives the bone tissue its rigid structure. We mentioned that blood is a complex specialized liquid tissue which contains a number of different cell types, and these cells are suspended in a liquid matrix. Now the whole blood, when we spin the blood in a centrifuge, we find that the blood cells will collect towards the lower half of the centrifuge tube, and this leaves behind a pale straw-coloured liquid above. Now this straw-coloured liquid is called plasma. Now this is a fluid which is made up of approximately 92% water, and you also find many dissolved substances such as nutrients, salts, hormones, and also large plasma proteins. Now you have to be careful here, because you might hear the term blood serum being used. Now blood serum is not the same thing as blood plasma. Blood plasma is essentially, as we've said here, the fluid component of the blood which is left behind when you remove the blood cells, and this includes the red blood cells, the white blood cells, and the platelets. When you remove those cells and cell fragments, the liquid that you're left with is called blood plasma. But if you then go on to remove the large plasma proteins, what you're left with then is a liquid called blood serum. So please don't mix up those two terms. Now, the white blood cells, when you spin blood in a centrifuge tube, we said that the red blood cells tend to collect near the bottom down here, and cells in general will collect in the lower half, but the white blood cells you'll find collecting at the top of the lower fraction. Now this consists, the white blood cells are of various different types. You have things like granulocytes, but also agranulocytes. You have lymphocytes, you have monocytes, and you have various other types of white blood cells. Now, there are between 5 and 10,000 white blood cells in every cubic millimeter of normal healthy blood. The red blood cells will form the majority of the lower fraction in the centrifuge tube, and these occupy about 45% of the total blood volume in a healthy individual. And there are between 5 and 6.5 million red blood cells in every cubic millimeter of healthy blood. Now, we go into the various types of blood cells in a lot more depth on that in that video on immunology, so if you do want more detail on that, I'd thoroughly recommend watching that video. However, let's just have a quick look at the different kinds of blood cells here. So this photomicrograph of a blood smear shows the appearance of blood when it's viewed under an optical microscope, and you can see here some of the different kinds of white blood cells, and you can see that they have different sizes and also different nuclear morphologies. Their nuclei are of different shapes and also sizes. So for example, you see here a B lymphocyte where the nucleus occupies the majority of the volume of the entire cell. Here you can see a phagocyte, which is sometimes referred to as a granulocyte, because you can see the cytoplasm has these granules in it. But you can see here that it has a very different nuclear morphology to, for example, a monocyte or a lymphocyte. You see here many of the red blood cells which have this biconcave disc shape, but you can also see these cell fragments called platelets, which are responsible, or involved rather, in the process of blood clotting as well. So we mentioned before that the plasma is the fluid matrix within which the blood cells and platelets are suspended. And you have three general categories of cells within the plasma. You have the red cells, which are sometimes called erythrocytes. You have the white blood cells, which collectively are referred to as leukocytes. And you have the platelets, which are referred to as thrombocytes. Now, lymphocytes, monocytes, and granulocytes, these are the different types of white blood cells. So let's move on to muscle tissue now. So there is skeletal muscle tissue and there's also cardiac muscle tissue. Now these are two very different types of muscle tissue. So the individual muscle cell fibers, which is the term used to describe individual muscle cells, you can see them here in this micrograph. So this would be one individual skeletal muscle cell that you're seeing here. And you can see that they are striated, they are striped in this manner. Now, these cells are specialized to produce contractile forces. They shorten and produce a tensile force. And collectively, all of those muscle fibers within a muscle are responsible for then moving the bones to which that muscle is attached by tendons at either end. Now, the exception for this is the skeletal muscle found in the tongue, which is only attached at one end. But in general, all skeletal muscle fibers are attached at either end to promote, to allow them to contract, to promote the motion of different limbs and other parts of the body. Now, the other thing about muscle cells is that they are multinucleate. Skeletal muscle cells are multinucleate. Each one of these muscle fibers actually contains multiple nuclei. Now, if you'd like some more detail on muscle fibers and the ways in which they contract, you can either watch one of the previous webinars in this series on the subject of control systems, but in that webinar we didn't go into muscle fibers in too much depth because that was explored in this video here that you can find on the YouTube channel on muscle physiology, where we go into muscle fibers and the ways in which they contract and their structure and functional features in a lot more depth. So if you're interested in muscle fibers and muscle in general, I'd advise you guys to have a look at that video there. And of course that video is also available on the front page of my website as well. Now, in contrast to skeletal muscle, cardiac muscle tissue has a very different cellular morphology. So cardiac muscle tissue is only found in the heart, and it's the tissue, the type of muscle which makes up the walls, the contractile walls of the heart. Now, unlike the linear skeletal muscle fibers, cardiac muscle cells are branched and they only have single nuclei. Now, the function of the heart is very different to the function of skeletal muscle. Skeletal muscle doesn't have to be constantly in operation through your entire lifetime. It's only really it's only really contracting when you want to move your limbs or other parts of your body, whereas your cardiac muscle has to be able to maintain a rhythmic contraction and quite a lot of force as well, and it's always contracting. It needs to be structured in such a way that it never gets tired. Now obviously that requires a different cellular structure. And so if we could magnify this region of the wall of the left ventricle, for example, so that we can examine the structure of those cardiac muscle fibers, we would see that they're very different in the way that they're structured and organized. So for a start, cardiac muscle fibers are uninucleate. Each muscle fiber only has a single nucleus, whereas these and they're also branched rather than linear. And as you can see also, they have these thickenings of the cell membrane where adjacent muscle fibers are in contact with each other, and these are called intercalated discs. So we have branched cells forming a network of contractile fibers. Now in this micrograph here we can see that as it actually appears under a microscope at about 1,000 times magnification. So here we can see 3 different muscle cell nuclei. We can also see the intercalated discs. If I go back here, we can see here the thickenings of the adjacent cell membranes between 2 muscle fibers forming these intercalated discs. Now the reason for this difference in structure is not just because of the efficiency and the longevity of the contraction of the muscle fibers of the heart, it's also because of the electrical properties of the heart. Now again, we go into that in a lot more depth in that webinar on control systems, where we talk about the control of the heartbeat. So again, if you want more detail on that, I suggest that you look in here. So let's look at nerve tissue, and again we go into nerves and neurons and the nervous system in a lot more depth in that control systems webinar. However, here we're just talking about the cellular features of nerve tissue. Now nerve tissue is made up of long thin cells which transmit nerve impulses, electrochemical signals around the body. Now these can actually travel rather long distances, such as for example between the brain and the legs. So here we can see the cell body of a motor neuron. We can see that the cell body has these branching dendrites which allow communication between this cell and adjacent cells, but it also has this long thin projection called an axon. Now this is a sensory neuron here that you're seeing here, and these are nerve cells as seen under an electron microscope. So all living things strive to efficiently undertake those 7 processes that categorize them as living things in order for the individuals and also the species to survive. So again, to remind you, there are 7 characteristic features of living things. There is respiration, the production of metabolic energy. There is movement. There is sensitivity to changes in their internal and external environments. There is the process of nutrition or feeding. There is excretion, which means the removal of waste substances from cell metabolism. There is growth and reproduction. Now in multicellular organisms, there tends to be a particular system which specializes in each one of these characteristic processes, and this is how the efficiency of these processes is ensured in multicellular organisms. Now, some scientists and authors now identify homeostasis, or the maintenance of an internal environment, as an 8th characteristic of all living things. So you might find that homeostasis can sometimes also be referred to as one of the characteristics that distinguishes living things from non-living things. Now again, in human systems, a system is made up of several organs and tissues that work together to perform related functions. So for example, the biceps, triceps and other skeletal muscles make up the muscular system of the body, and this enables movement and locomotion. We have the stomach together with the other organs of the gastrointestinal tract, such as the liver and the small intestine, for example, which form the digestive system. And we have the heart together with blood vessels forming the circulatory system. We have the bones and of the skull and the ribs and other bones of the body, which together will form the skeletal system. Now, sometimes the skeletal system and the muscular system are referred to collectively as the musculoskeletal system, because really neither of them can function without the other. We also have the nervous system, which is made up of the central nervous system, composed of the brain and the spinal cord, and the peripheral nervous system, which is made up of the cranial and the spinal nerves. We also have the ovaries together with the uterus in the female, which form the reproductive system, and obviously in the male we have the testes, which form the equivalent. And we have the lungs together with the airways, like the trachea and the bronchi and bronchioles, and the diaphragm, which form the respiratory system. So we also have the endocrine system. Now the endocrine system is the collection of all glands which secrete hormones, endocrine glands. So the endocrine system is the system of glands and organs which produce hormones. Now, the human organism is made up of all of these systems collectively working together to perform all 7, or if you include homeostasis, all 8 of those functions. And of course we can refer to other systems as well, such as the renal system, but these are just given as examples here. So we've talked about tissues and organs in animals, but we also need to be aware of tissues and organs in plants as well. So, as in animals, the division of labour to perform all 7, or again, in counting homeostasis, 8 of those functions of living organisms, that division of labour can also be seen in plants in the way that cells can be organised into tissues and tissues into organs. So multicellular plants are organisms which are made up of thousands or millions of specialized cells, which are grouped again into tissues, tissues grouped into organs, organs grouped into systems. So like animals, plants have levels of organizations based on that scheme. And again, there are 6 principal kinds of plant tissues which you should be aware of. So there are photosynthetic tissues such as the spongy mesophyll and the palisade mesophyll that you find in leaves. There is the epidermal tissue, and this is kind of equivalent to the epithelial tissue found in animals. So in plants, the epidermal tissue is the outer layer tissue of the parts of the plant, such as the root hairs and the epidermis. You have the vascular tissues for the mass transport of substances, such as the xylem and the phloem. Now we're going to get into the vascular tissues, the xylem and the phloem, in a lot more depth in the final webinar of this series on the subject of the exchange and transport of substances. There's also the meristem tissue, which is the tissue found in the growing regions of the plant, such as the cambium tissue. There's packing tissue, which is, so for example, there's the parenchyma tissue, which is responsible for bulking up the stem and the roots. And finally, there's strengthening tissue, such as collenchyma, which has some thickening of its cell walls, and also sclerenchyma, which is usually dead tissue, which has thick, lignified secondary cell walls. And these are the tissues which provide the main structural support and strength to the multicellular plant. So let's look at these tissues in a little bit more detail. And again, we go into examples of these in a lot more detail on that course module on cell biology on my website, but we're just going to kind of quickly look at the main features of each of these types of tissues here. And once we've done that, I'll give you guys another 5-minute break before we move on to the next section of this webinar. So, for example, we have the photosynthetic tissues, which examples of which are the palisade and spongy mesophyll layers in the leaf. So, you will have seen diagrams like this all the way from GCSE biology onwards, or perhaps even earlier. This is a section through a leaf as viewed under a microscope. Here we can see the palisade mesophyll layer. These are the palisade cells here, which are arranged in this columnar fashion, and they contain a high density of chloroplasts. Here we can see the spongy mesophyll layer. Now, these cells are surrounded by a thin film of water and have these air pockets between them to facilitate efficient gas exchange. So the presence of various tissues, such as the palisade and spongy mesophyll layers of the plant, these will define the leaf as a plant organ. So leaves are the organs of the plant whose function is photosynthesis. So if we take a transverse section through a dicotyledonous leaf, we can see here the mid-rib of a privet leaf from this organism, Ligustrum. And this is typical of a dicotyledonous leaf. Now again, we're going to explore the features of dicotyledonous leaves in the context of exchange and transport in the next webinar of this series. But here we can see the lower epidermis. Here we can see the parenchyma cells. These are the intercellular air spaces in the spongy mesophyll layer. These are the spongy mesophyll cells. These are the palisade cells here. And this is the upper epidermis layer here. So we can see various tissues here in the midrib of a leaf, and also in the transverse section of the gas exchange portions of the leaf as well. Here in the central portion we can see the xylem tissue. We can also see the stoma and the guard cells. And here we can see the phloem vessels as well. So if we were to take a section through here and magnify it further, this is what we might see under a light microscope here. Now again, this is typical of a dicotyledonous leaf from any species. So here we can see the upper epidermal layer. Here we can see one of the vascular bundles. Here are the columnar palisade cells with their high concentration of chloroplasts. Down here we can see the spongy mesophyll layer, and here we can see, this label is slightly off, apologies for that, here, and this label as well, but here we can see the guard cells and the opening called the stoma. These are the spongy mesophyll layer cells here and here, and you can see in the spongy mesophyll layer these air pockets very clearly here. Here we can see a pair of guard cells which have closed this stoma over here. And down here we can see the lower epidermal layer as well. Now, a couple more points, interesting points about palisade cells before we go any further. So if we look at a single palisade cell here, now palisade cells, like all cells in a multicellular organism, are very well adapted to their role, and their role is as the principal photosynthesizing cells of the leaf. Firstly, they have a column shape like this. Now this ensures that the cell allows for close packaging of these adjacent cells close to the surface of the leaf, and each cell possesses numerous chloroplasts. Now the chloroplasts are actually able to freely move around inside the cytoplasm. Now the reason for this is that they will orient themselves to maximize the interception of sunlight energy. So the chloroplasts have the ability to orient themselves in positions that maximize the light absorption in dim light, and also to orient themselves to reduce the amount of exposure to light if the light intensity gets too high and is in danger of bleaching the chloroplasts. So here we can see the cell membrane, the cell wall, the tonoplast containing the cell sap making up the large central vacuole. Here we can see the chloroplasts, and here we can see the nucleus of the cell. So palisade cells have many chloroplasts in them as compared to other cells, including the spongy mesophyll layer. Now again, if you think about it, the palisade cells are there on the upper surface of the leaf, just under the upper epidermis, and as such they are the first cells to intercept the sunlight energy which is coming from the sun. Now these are very well suited to their photosynthetic role, as we said before, because first of all they have this columnar shape. Now the columnar shape means that the light has to travel through the maximum number of chloroplasts in order to get through that layer of cells. But also the chloroplasts are able to move freely around in the cytoplasm. Now under conditions of dim light, they will collect in the upper surface of the palisade layer nearest the upper epidermis, like this. Now this is to, and again they will orient themselves like this to give the maximum surface area and intercept the maximum amount of light energy, because they're under conditions of dim light. Now if the light gets too bright and is in danger of damaging the chloroplasts, they will change their orientation by moving to the opposite end of the cell. And in this case, they don't orient themselves in a horizontal orientation like this, they orient themselves in a vertical orientation like this, and this is to minimize the chance of the intense light damaging the chlorophyll and bleaching it. So let's look at the epidermal tissue now. So the epidermal layer protects the tissues that lies beneath it, and the epidermal cells will secrete a waxy layer over the surface of the leaves, and this is basically for waterproofing. So the epidermal layer is usually one cell thick, as you can see in this diagram, and this includes the lower epidermal layer as well. So here we can see a micrograph, a photomicrograph of onion epidermis, and many of you will have seen this in the course of your required practicals. Now the epidermis of the roots actually has these finger-like root hairs that project outward from it. Now the roots of the plants are the organs which are responsible for absorbing water and mineral ions, dissolved mineral ions, from the water in the soil, and as such these root hairs provide the maximum surface area for that absorption to take place. So if you look at the root of a plant, it might appear like this, with this very fine coating of hairs which themselves have tiny microscopic root hairs coming from them. So the root hairs are actually modified epidermal cells, and these create again a very large surface area for the efficient absorption of water and mineral ions from the surrounding soil. Now we mentioned that the leaves have these pores in their undersides called stomata, and again stomata is the plural, the term stoma is the singular term. So if we were to magnify the underside of this leaf here, we might see something like this. So here we can see the guard cells viewed directly face on, and the pores are called the stoma. So these allow the carbon dioxide needed for photosynthesis to enter the leaf from the atmosphere, but also the oxygen being generated by photosynthesis, which is not being used by the plant cells for respiration, the excess oxygen diffuses outwards into the atmosphere through the stoma when they're open. So each stoma is a pore between two guard cells in the epidermis. Now the guard cells can change their shape in order to close or open the stoma. So the stomata are essentially the sites of gas exchange between the leaf and the environment. Now these are the pores that allow for the exchange of gases, and their size is regulated by these specialized cells that we call guard cells. Now the stomata are found in the lower epidermis of leaves, although they can be present in fewer numbers in the upper epidermis as well. Now, the large number of the stomata, together with the regulation of their size by the guard cells, this allows for the efficient gas exchange, the exchange of oxygen from the internal compartment of the leaf to the external atmosphere, and the opposite diffusion of carbon dioxide in the opposite direction from the atmosphere into the leaf, where it can be used by photosynthesis. So here is the lower epidermis of a tulip leaf, shown under a magnification of 400 times as viewed under an optical microscope. Here we can see the thick inelastic inner wall of the guard cell, and we can also see, apologies for this being slightly off, the thin elastic outer wall of the guard cell. So this is the thin elastic outer wall, this is the thin elastic outer wall and the thick inelastic inner wall. And you have the stomatal pore here. Again, apologies for these labels being off. I think it's probably a function of my PowerPoint going a bit wonky. The joys of live broadcasting. So these would be the epidermal cells, and you can see that there are many stomata embedded between these epidermal cells. So you see these pairs of guard cells with the stomata between them, and these are numerous on the lower epidermis, although you can find them occasionally in far fewer numbers on the upper epidermis as well. Now, if we view these under an electron micrograph, this is what they would look like. So this scale bar gives you an indication of the size here. Here we can see this stoma is open, and through the open stoma we can actually see one of the mesophyll cells beneath that stoma. So we've come up to the end of the second hour of this webinar, so I'm going to give you guys another 5-minute break. After the break, we're going to get into the subject of prokaryotic cell structure and also the structure of viruses. Let me share my screen again with the Zoom webinar attendees here. Okay. Alrighty, so prokaryotes. Let's start with prokaryotes. Now, we mentioned before that in general, in modern biology, even though this is something that is continually under revision and under discussion amongst biologists, in general, we classify organisms before we start to classify them into the kingdoms, phyla, classes, orders, family, genera, and species. We classify them first as either being belonging to the domain of the bacteria, the domain of the prokarya, or the domain of the eukarya. Now, bacteria and archaea are prokaryotic organisms, so let's have a look at their structures in a bit more detail. So prokaryotic living things belong to two domains, the bacteria and the archaea. So if we look at those in a bit more depth here, so here we see a photograph of bacterial colonies growing on nutrient agar in a petri dish. Now, again, like eukarya, prokarya are a diverse group of organisms, and they occupy an even wider range of habitats than eukarya, and again they display various modes of nutrition. Now, members of these two domains typically obtain their nutrition either by absorption, by photosynthesis, and also by chemosynthesis. Now, saprobiotic bacteria, photoautotrophic bacteria and chemoautotrophic bacteria respectively are the types of the bacteria organized or classified in terms of their mode of nutrition. So again, the idea of autotrophy or self-feeding, this can occur in a number of ways. Eukarya, the domain eukarya, contains a number of a couple of kingdoms where you have autotrophic organisms. So for example, the kingdom Plantae, these are autotrophs which obtain their nutrients by a process of photosynthesis. So we would describe these as photoautotrophs. Now, the Organisms that you find in these two domains, the domain of the bacteria and the archaea, these are also autotrophic organisms for the most part, but they can also sometimes make use of chemical processes in their environment in order to generate the energy that they need and to generate the nutrients that they need. And this would be described as chemoautotrophy. So the vast majority of the members of these domains are unicellular organisms, although sometimes they can organize themselves into colonies. So for example, blue-green algae can form aggregated cells, and these can form chains and other types of colonies as well. Now, specialized cells within these aggregates are able to fix nitrogen. So, reproduction in prokaryotes is mainly by asexual means, by a process which resembles cell division in Eukarya, but is very different in the way that it occurs. Now, this is called binary fission. Now, unlike eukaryotes, prokaryotic organisms such as bacteria can transfer genetic information and characteristics between each other horizontally rather than vertically. And what I mean by that is vertical transmission of genes and characteristics. This describes the process by which one cell will transfer its genetic material and characteristics to daughter cells by cell division. But eukaryotic organisms are unable to transfer genetic material between individuals. Now, this is the process of horizontal transmission, but this is possible in prokarya. Now, all cells among the prokaryotic domains possess typical features. So the feature that distinguishes prokaryotic organisms from eukaryotic organisms is the lack of membrane-bound internal organelles. So prokaryotic organisms like bacteria do not have internal organelles which are surrounded by membranes. Now, another feature which distinguishes these organisms from eukaryotic cells is that the chromosome is non-linear. It's not a linear, it's not based upon a linear molecule of DNA. Instead, the DNA molecule in a bacterium, for example, is a continuous loop. So the chromosome is a large loop of genetic material made up of DNA. Now, in bacteria, again, the cell wall that you find in bacteria is made up of a different kind of material than the cell wall that you find in plants or fungi. In bacteria, the cell wall is made up of murine, and there is no murine in the cell walls of the archaea. Now, the archaea, in contrast, have a different kind of cell membrane as well in terms of its biochemical constituents. So the archaea have membranes made up of glycerol ether lipids. Now, bacteria and eukaryotes have membranes which are made up of glycerol ester lipids, in other words, phospholipids. Now, the archaea have more complex forms of the enzyme involved in protein synthesis than you find in bacteria. Now, in the archaea, their genes and their method of protein synthesis are more similar to eukaryotes than found in bacteria. So, for example, one of the environments in which you find these organisms are the hydrothermal vents that you find in the floor of the ocean, such as in the Pacific Ocean bed. So, an example of a food chain that you find these organisms in. So, in these environments, there is no light available. The light that you see here reflecting off this hydrothermal vent is artificial light from a submersible robot which is taking this photograph. But under normal conditions, this environment is in complete darkness. So the organisms down here cannot make use of photosynthesis to power their metabolism and to produce their nutrients. Instead, they make use of chemosynthesis. So what you have here is superheated water under enormous pressure, where the water can reach temperatures in excess of 400 degrees centigrade and not boil because it's under such a high pressure. Now, the water coming out from these hydrothermal vents is actually coming from deep within the Earth's crust, where it picks up many different minerals from the chemical reactions occurring down in the crust of the Earth. And these include things like hydrogen sulfide. Now, hydrogen sulfide is the chemical which is responsible for the rotten egg smell that you can smell when eggs will decompose, for example. So hydrogen sulfide from the Earth's core, found in this superheated water emerging from this hydrothermal vent, this chemical is used by archaea and chemosynthetic bacteria, and these form the primary producers of this food chain. Now these organisms will make use of this chemical to generate simple sugars. Now in turn, these primary producers are the food of the primary consumers within the food web in this environment, and these include things like barnacles, clams, anemones, mussels and small worms. Now these primary consumers are in turn fed upon by secondary consumers such as large crabs and fish, which you also find in these environments. Now all of the living things that you find in these deep sea ecosystems are actually called extremophiles. Now the term extremophile refers to the fact that these organisms seem to thrive in extreme conditions. So for example, high temperatures, thermophiles, and also the high pressures that you find down here. Now again, we go into these ecosystems and these processes in a lot more detail in that course module on cell biology on my website, but for the purposes of just of brevity and summarizing things here, this is about as far as we'll go for the moment in this webinar here. So let's look at the general structure of a bacterium. So this is the general structure of a rod-shaped bacteria, but bacteria come in all different morphologies. They can be rod-shaped, they can be spherical shaped, they can even be spiral shaped as well. And again, we're not going to go into that in too much depth here. We're just going to look at the generalized structure of this type of bacterium here. So there are structures which you find in all bacteria, but there are also structures that you find in some bacteria. So let's look at the structures that you find generally within this domain. So you find a cell wall which is made up of murine. Obviously you will find a cell surface membrane underneath that. You'll also find these food reserve granules. You find the circular looped chromosome made up of DNA, and this again will float freely in the cytoplasm because prokaryotes do not have a internal membrane bound organelles, and so they don't have a nuclear envelope around their chromosome. You also find these smaller 70S ribosomes, and of course you also find the cytosol in which all of these are suspended to form the cytoplasm. Now these are always present in all bacteria, but you also find some other structures which are sometimes present in some types of bacteria. So these include these finger-like projections which are called pili or fimbriae. You find sometimes these smaller circular loops of DNA called plasmids, and it's these plasmids which can be exchanged between individuals to transfer beneficial characteristics between individual bacteria. You can also sometimes find around the cell wall of some bacteria an additional slime capsule. Sometimes this is called the slime layer, and this is essentially to protect bacteria from drying out in dry environments, but also can protect them from environmental chemicals which could potentially damage them. Some bacteria also have a long projection, a tail-like projection called a flagellum, sometimes more than one, and again flagellum is the singular, flagelli is the plural. Now the flagellum is very, very interesting. It's the only freely rotating structure found in biological systems. This will literally whir around, it will whiz around and around like a little propeller, and its function is to propel this bacterium through the fluid in which it finds itself. So this is an electron micrograph of E. coli, and this shows the flagellae of this bacterium very clearly. So this is the bacterium, this is the form of E. coli, E. coli 0175. This is what causes food poisoning in many cases. Now there are hundreds of different strains of E. coli, and most of these are actually harmless and normally will be found in the healthy human and animal intestines. But this particular strain of E. coli produces a very potent poison, a toxin, and it's this toxin that causes severe food poisoning. So bacteria are prokaryotic, unicellular living things, although they can stick together and form pairs, clusters and chains. So let's quickly summarize the general features of bacteria. They are typically smaller than eukaryotic organisms. They range in size from between 0.5 and 10 micrometers in length. They are unicellular organisms. They reproduce by a process of binary fission. And they possess a circular DNA molecule which lies naked and without an envelope in the cytoplasm. As prokaryotic organisms, they lack membrane-bound internal organelles, and they possess a rigid cell wall with a multi-layered structure made up of a different material than that found in eukaryotic cell walls, and it's a material called murrein, which is a type of peptidoglycan. They also possess smaller ribosomes than are found in eukaryotes, and they display infoldings or invaginations of the plasma membrane known as mesosomes. Now this is where the respiratory reactions take place. They sometimes have numerous fimbriae or pili which project from the cell surface. Now the pili allow for adhesion to surfaces and also sticking to other cells. And these are involved also in the reproductive process of conjugation in bacteria. They also sometimes have flagellae, and these have a simple structure and are used in locomotion. Now we mentioned that the genome of bacteria consists mostly of a circular DNA loop which forms the bulk of a region of the cytoplasm called the nucleoid. Now the nucleoid is the equivalent in a bacterium of the nucleus. So remember, bacteria do not have internal membrane-bound organelles, and so the bacterial chromosome it does not have a nuclear envelope around it, but it is clustered and coiled and folded into a region of the cytoplasm which is called the nucleoid. Sorry, I had to mute the microphone there in order to cough and not deafen you all. So, in addition to this bacterial chromosome that's found in the nucleoid, you also sometimes find these smaller loops of DNA called plasmids. Now, the plasmids are able to replicate themselves independently of the bacterial chromosome, and they carry sometimes additional genes which can be passed on to other individuals by horizontal gene transmission. Now, these sometimes are in the example of bacterial resistance, when one bacterium of a population develops the resistance to a particular antibiotic, that gene which confers that advantageous characteristic can be passed to other individuals by these plasmids. So we also mentioned that the bacterial ribosomes are smaller than those found in eukaryotes. So we find 70S ribosomes in contrast to the 80S ribosomes which are found in eukaryotes. So to summarize, we say that we can summarize the features of prokaryotic cells here. I'll leave that up on the screen while I take a sip of water. Okay, so we've talked about the general features of prokaryotic cells. We've also talked about the general features of eukaryotic cells. Now, viruses are acellular. They are not made up of cells, and in fact, many biologists would not classify them at all as being living things. However, the specification does require you to have a knowledge of the structures of viruses as well. And so we're going to talk about the structural features of viruses, and we'll also talk about the general manner in which viruses replicate themselves, because viral replication is really the only feature which resembles the characteristics of living things. So let's have a look at the way that viruses are classified. Now, no matter what system of classification we use, viruses are classified as being acellular. In other words, they are not made up of cells, and as such they are non-living, so they don't feature in either the 5-kingdom or the 3-domain systems of classifications. So if viruses are not living things, then what exactly are they? Well, they can be thought of simply as self-replicating particles which can be found in the context of biological systems. Now, virus particles, or virions as they're sometimes called, these are small infectious agents which use the metabolic pathways of living cells to replicate themselves. So viruses consist of 2 or 3 structural parts. Firstly, we have the genetic material of the virus. Now, depending on what kind of virus you're looking at, that genetic material can either be DNA or RNA. Around the genetic material of the virus, you find a protein coat or a capsid, and this has the function of surrounding and protecting the genome of the virus. Now, in some virions, such as herpes, pox, and HIV viruses, the capsid is itself surrounded by an envelope when the virion is found outside of cells. Now, the envelope itself actually consists of a phospholipid bilayer and some of the membrane proteins from the host cell. Now, this is what allows those viruses to evade the normal defenses of the immune system. Now, again, if you want some more details of that, I'd advise you guys to have a look at that video on immunology. So, the envelope that surrounds, for example, the HIV virus consists of the bilayer, the lipid bilayer, which is derived from the membrane of the host cell in which you find the virus. Now, this essentially has the function of hiding that virus from the cells of the immune system and from the antibodies produced by the immune system, because all of the external surface of the cell of the virus now is made up of proteins and phospholipids which come from the host organism's own cells. And so when the antibodies and the cells of the immune system encounters this enveloped virus, they can only detect the proteins and the phospholipids and other molecules and antigens which are normally present on the cells of that organism. And so it's very difficult for the immune system to detect and mount an efficient response against these kinds of enveloped viruses. Now, also, viruses like HIV are referred to as retroviruses because they have the enzyme reverse transcriptase. Now, this enzyme allows them to inject their own DNA into the genome of the host organism. So here we can see the envelope of the virus. We can see around this envelope, embedded within this envelope, the glycoproteins that you see here. Within the capsid of the virus, you can see the RNA strands. Again, this is a diagram of HIV. And HIV is an RNA virus. And around the virus you can see the protein coat, or the capsid, and within there you can also find molecules of the enzyme reverse transcriptase. Now, all viruses by definition are infectious. They can cause many diseases of animals, plants, and even bacteria. Now, they can be very resilient outside of the host, and some can be dried into a powder and stored for many years, but only become active when they become hydrated again under conditions where sufficient water is available, either outside of cells where there's sufficient water or within cells and tissues where there's a fluid environment. So here we can see a couple of different examples of viruses, and you can see that they all show very different shapes and morphologies. So, viral diseases caused by viruses in humans include things like the common cold, influenza, smallpox, hepatitis, polio, rabies, and of course, AIDS. Now, some viruses are also thought to be implicated in the development of various kinds of cancer as well. So, viruses can be thought of in some cases as carcinogens, agents which can cause cancer. Here we can see a diagram of a flu virus, in this case the one that causes bird flu. So, unlike other organisms, unlike living organisms rather, viruses really only perform the function of replication or reproduction. They don't respire, they don't feed, they don't do any of the other 7 or 8 characteristics of living things. Now, the way that viruses operate is they effectively hijack the normal cellular machinery of transcription and translation in order to build new viruses inside infected host cells. So essentially, they turn host cells into virus factories which produce more viruses. So after as little as 30 minutes, the host cell will burst open to release new viruses which go on to infect other host cells. So this animation here, if I bring it up on the screen for you, this illustrates the life cycle of a bacteriophage virus. Now, bacteriophage viruses are the viruses which infect bacterial cells. So here we have the bacterial cell represented by this diagram. This red loop here represents the bacterial chromosome. So to begin with, the virus infects and attaches itself to the cell surface, the cell wall. It injects its own DNA or RNA into the host bacterial cell. At this point, the viral DNA will combine with the bacterial DNA and be spliced into the bacterial genome in this manner. So this blue segment here represents the viral DNA, which has now become spliced into the bacterial genome. What will happen now is that viral DNA is transcribed and translated into the protein components of new viruses, and also the viral DNA itself will be replicated to form more viral DNA. So essentially what's happened here is the virus has hijacked the normal cellular machinery which is responsible for producing proteins and replicating the DNA of the bacterial cell to produce viral DNA and the protein components of the virus. Those components are then assembled to form new viruses until eventually the cell will burst open, and this releases those new viruses into the environment where they can infect more host cells. And the process is very similar when viruses infect eukaryotic cells as well. So the animation that you've just seen combines two different methods of bacteriophage replication, the lytic and lysogenic cycles. And if you want more detail on that, again, by all means, check out that course module on my website on cell biology. So the next thing that we have to look at in the specification is the different ways in which biologists will study cells. So we're going to look at the principles of optical and also electron microscopy, and we're also going to look at the ways in which the cell components are broken up and separated for further study using microscopy, and this includes cell fractionation and cell ultracentrifugation. So let's start with optical microscopy to begin with. Now, before we go any further, we need to mention a few things about units of measurement. Now, for some reason, many students will have difficulties with conversion between different units. Now, you need to be aware that scientists in general, and again, apologies for the sounds of cars and things honking in the background. Again, it is very, very hot here in London, and I've had to open the windows. Again, please do let me know, those of you who are in attendance, if that does become too distracting. So, we need to be aware that sometimes you will see prefixes in front of certain units in science. So the prefix milli or micro or nano or pico or femto, these all refer to orders of magnitude of measurement. So typically when we're studying things under microscopes, we will normally be measuring their lengths and sizes in units of micrometers or nanometers. Now, micro refers to 1/1,000,000th of a particular measurement. So a micrometer is 1/1,000,000th of a meter, whereas a nanometer is 1/1,000,000,000th of a meter. And you need to be comfortable with interconverting between these units. So keep in mind that 1 meter contains 100 centimeters, so the prefix centi means 100. The prefix milli means 1,000, so centi means 1/100th of a particular measurement, so a centimeter would be 1/100th of a meter, so there are 100 centimeters in a meter. Similarly, if you talk about a centiliter, well, that would be 1/100th of a litre, so that would be equivalent to 10 milliliters, for example. Now, 1 millimeter is 1,000th of a meter, so if you're converting between millimeters and centimeters, 1 centimeter will contain 10 millimeters. 1 micrometer, and micro, the prefix is given the symbol mu, the Greek symbol mu, which looks like a little U with a tail on it. So if 1 micrometer is 1/1,000,000th of a meter and 1 millimeter is 1,000th of a meter, this means that 1 millimeter contains 1,000 micrometers. Now, 1 nanometer is 1/1000th of a meter, and so 1 micrometer contains 1,000 nanometers. So please be careful and please ensure that you're comfortable in your knowledge of what these units and these prefixes mean, and that you're comfortable with converting between these units, because this is typically a source of a lot of errors in exam questions. So let's look at some key terms here. So we first need to be aware of the distinction between the term object or specimen and the term image. Now, the object or specimen is the actual material object that you are viewing under the microscope, whereas the image is the appearance of that object as viewed under that imaging device. So, for example, if you're viewing a Daphnia, a tiny aquatic crustacean, which looks like this, under a microscope, the Daphnia would be the object or the specimen, and the picture that you observe down the microscope would be the image of that object or specimen. So, the modern optical compound microscope or light microscope is essentially an instrument that makes use of a series of lenses, convex lenses, through which the light can be focused to obtain an enlarged image of the specimen. So this diagram here shows the series of parts of the microscope through which the light travels in its path from the source of light to your eye. So you have a light source, which in the case of an optical microscope is typically either a bulb or external light, which has been reflected onto the specimen stage. You then have a glass condenser lens. Now the condenser lens is not designed to magnify the image because of course the light hasn't yet transferred through the specimen. The function of the condenser lens is to focus the incoming light intensity onto the image, onto the specimen. You then have the specimen, which is on a slide. Now the light will be transferred through the specimen and into the objective lens. So the objective lens is the first lens which is responsible for the magnification of the image. You then have the eyepiece lens, which is responsible for further magnification and focusing of that image. So the three lenses that make up the optical system of a typical light microscope are the eyepiece lens, the objective lens, and the condenser lens. Now remember, the condenser lens does not contribute to the magnification of the image. The function of the condenser lens is simply to collect the incoming light intensity and focus it onto the specimen to produce a brighter and better image. So if we're looking at the parts of an optical microscope, we firstly have the eyepiece, which is used to view the image formed after the light has passed through the objective lens. And again, the path of light in this case is from here upwards through the condenser lens, through the objective lens, through the eyepiece lens, and into the eye. Now you have two focusing knobs here. The first is called the coarse focus, the larger of the two knobs. The second is called the fine focus. You have the objective lenses, and usually there's more than one of these, typically about 3 of these, each of which has a different power of magnification. You have a stage for positioning the slide on which you have the object or the specimen. You have clips to hold the slide in place. You also have a condenser unit, which is used to control the amount of light entering the objective lens. You have a lever for controlling an iris diaphragm, which can be adjusted to alter the amount of light entering the objective lens. And typically you'll either have a concave mirror or a bulb as the source of light. Now a concave mirror is used in this case to reflect light from the environment onto the condenser unit, which can then pass that light through the specimen stage. So we need to define a very important term, resolution, here. So the most important factor in obtaining a good image from the microscope is the ability of the objective lens to reproduce detail in the image. Now the ability to reproduce detail is known as the resolving power or the resolution of the microscope being used. Now the definition of resolution is that it is the smallest distance between two particles which allows them to be distinguished from one another in the image produced by the microscope. Now the unaided human eye has a resolving power of about 0.1 millimeters on average. So in other words, your eye, with no magnifying aid of a microscope or any other instrument, it can distinguish between two lines or two dots separated by 0.1 millimeters as being two distinct objects. But the resolving power of a light microscope is much higher than that. It's 200 nanometers, whereas the resolving power of an electron microscope is higher still at 0.2 nanometers. So objects closer together than 0.1 millimeters apart cannot be distinguished by the average unaided human eye and typically will be seen as a single object. So the resolution of the light microscope is limited by the wavelength of light which is being used. Now magnification in contrast to resolution, is not dependent upon the resolving power, but as magnification with the optical microscope increases, the limited resolving power gives blurred images. Now, the very best optical microscope operate at a resolving power of approximately 0.2 micrometers with a magnifying range of between ×5 to ×1500. So what we're saying here is resolution, the ability of a device like an optical microscope to distinguish objects between each other, is defined as the minimum distance of the separation of two lines or two dots where that instrument can still produce an image where those two objects are still distinguishable from each other as discrete objects, whereas the magnifying power is simply the power of a device to magnify an object to a particular size of image. So let's look at magnification now. So different magnifications with an optical microscope are achieved by choosing different objective lenses and also by choosing different eyepiece lenses. So focusing, on the other hand, is achieved by turning the coarse and fine focusing knobs. So let's look at how magnifying power is achieved in a microscope. So firstly, remember, we have two sets of lenses which are both capable and both responsible for magnifying the image. The first of these is the eyepiece lens. Now, eyepiece lenses in modern optical microscopes will range in magnifying power from ×5 to ×15. Now, there's a much wider range of objective lenses available in optical compound microscopes. Now, the most common of these that you may well have come across in the laboratory are things like ×5, ×10, ×20, ×40, usually up to a maximum of ×100. Now, if you're using ×100 lenses, these are usually used with an immersion oil. So usually if you're using the higher power of objective lenses, what you'll first do is place a very small dot of oil on the surface of the cover slip and then lower that lens to the point where the objective lens and the cover slip are connected by that oil dot. Now, the reason why we do this is that this excludes air from between the specimen and the cover slip over the specimen and the objective lens. And the reason that we do this is just that small distance of air and the particles floating through that air at that high magnification, that can blur the image. And so to prevent that from happening, we use a dot of oil to connect the surface of the objective lens and the cover slip. And this produces a clearer image. Now, the total magnification of the device, the microscope, is calculated by multiplying the magnifying power of the eyepiece lens, or ME for the magnifying power of the eyepiece lens, times the magnifying power of the objective lens, or MO for short. So the total magnification achieved by an optical microscope is the product of the magnifying power of the eyepiece lens multiplied by the magnifying power of the objective lens. So the modern optical compound microscope is essentially an instrument that uses a series of convex lenses through which light is first focused in the case of the condenser lens and then magnified by the objective lens and the eyepiece lens to produce an enlarged image of a very small object. And again, there are two lenses which contribute to the magnifying power, the eyepiece lens and the objective lens. So let's have a look at how we calculate magnification using a couple of examples here. Now, the standard eyepiece lens is ×10 magnification, but there are a range of objective lenses with different magnifications. So for example, you can have a ×10 objective lens, which you can see here, or you can have a ×40 lens. Now, typically, the higher the magnification of the objective lens, the smaller the diameter of those lenses. So to calculate the size of a specimen on a microscope slide, you have to use the slide with an etched graduations on it, and these are this is called a micrometer slide, and also a special insert for the eyepiece, which is called a graticule. So the micrometer slide, if you were to magnify it, you would see a a very small etched graduation here indicating specific distances here. So 100 units along the micrometer slide is equivalent to 10 millimeters. Now, the micrometer slide is then placed on the stage, and the graticule is then inserted into the eyepiece. Now, the eyepiece graticule contains another of these etched graduations. So how do we use the micrometer slide and the graticule to calculate very accurately the size of an object as viewed under the microscope? Well, for a start, the scale on the stage micrometer here is first put onto the slide and then brought into focus using the coarse and the fine focusing knobs so that it's clear on as viewed through the eyepiece like this. And then it's moved to overlap the graticule on the eyepiece, which is shown here. Now, in the following example, 100 of the smallest measurements on the graticule, corresponding to 0.1 millimeters, are equal to 3 millimeters on the micrometer or specimen slide at a particular magnification. So in this particular example, 100 graticule units is equivalent to 3 millimeters. Now what this means is that one graticule unit is equivalent to 0.03 millimeters, and therefore the size of the specimen will be however many units the specimen measures across as viewed through the eyepiece, multiplied by 0.03 millimeters to give the size of the specimen in millimeters. So this is one of the examples that appears in your student revision and workbooks that accompanies this course module or this webinar for those of you who booked tickets in advance. So a slide of some onion cells was prepared and observed under a microscope at high magnification, and we want to know what is the diameter of a nucleus of one of these cells. So again, in this example, 100 graticule units is 0.3 millimeters, and this makes one unit equivalent to 0.003 millimeters. Now, if we, again, according to this single unit of measurement, this means that the size of any specimen as viewed under this particular microscope set up in this way is going to be however many units that object measures multiplied by 0.003 millimeters. So if we're looking at this nucleus, for example, we can see that the nucleus measures 5 graticule units along here, and therefore we simply multiply 5 units by 0.003, and this tells us that this nucleus has a size in actuality of 0.018 millimeters. So this photo of some onion cells was taken at a magnifying power of 200 times, and again this is another example that appears in your student workbooks. And again, if you haven't already uploaded your completed student workbook with your answers up to your private coaching area, please do so, and I'll be happy to provide marking and feedback on that for you. So in this example we want to know the actual length of one of these onion cells. So in this case we have a photo produced of from the microscope. So we have a physical photo in front of us on the desk. So in this case, the way that we would do this is we would use a ruler to measure the length of one of these onion cells on that photo in millimeters. So we might do that like this. And we find that this cell's length as measured on the photo by this ruler is 60 millimeters. So again, using the formula, which you should all be familiar with, this is how we calculate magnification. The size of, or rather the size of the object. So the size of the object is the size of the image divided by the magnifying power. And of course you can rearrange this because magnification under any conditions with any microscope is always defined as the size of the image divided by the size of the original object. So if we rearrange that to find the size of the object, we get this. We then substitute our values of the size of the image divided by the magnifying power, and we find that the actual length of this cell is 0.3 millimeters. So another example that appears in your revision and study packs for this webinar is this one. So this photo shows some human cheek cells taken at a magnifying power of 500 times, and we wish to know the actual length of one of these cheek cells. So again, the way that we would do this is, again, we would measure the size of one of these cells on the photo using a ruler. We have the magnifying power, and so we can simply substitute these values into the formula. We evaluate and we find that the cell is 0.06 millimeters in diameter. So we've looked at optical microscopy. We also need to understand electron microscopy. Now, an electron microscope obviously looks very, very different to a light microscope, as you can see here, and it works in a very, very different way. Now remember, the light microscope, the resolving power of a light microscope is limited by the wavelength of light that you're using to illuminate your sample. Now, in the course module on cell biology, in this lesson that deals with optical and electron microscopy, we go into a lot more depth of a couple of other types of optical microscopy, for example, confocal and fluorescence microscopy. We're not going to go into that in detail here, but those types of microscopy allow us to see higher resolution and higher magnification than a conventional optical microscope. But nonetheless, even those types of more advanced optical microscopy are limited in the sense that the resolving power of any optical microscope is limited by the wavelength of light. So even if you use the shortest wavelengths of visible light, the blue wavelengths, you're still limited by the fact that those wavelengths have a specific length of the wave. Now, if the two objects which you're trying to distinguish as two different objects are closer together than the wavelength of light that you're using to illuminate those objects, then that optical microscope will be unable to resolve those as different objects. And so optical microscopes, as we said before, regardless of what type of microscopy you're doing with light, you are limited by the fact that if the objects that you're trying to resolve are closer together than the wavelength of light that you're trying to use to illuminate them, you cannot distinguish them as separate objects in the image. What you need in those circumstances is an electron microscope. Now, electrons have a much shorter wavelength if you model them as waves than a single wavelength of even blue or violet visible light. And so if you use electrons rather than light in your microscopy, you're able to resolve to a much higher degree of resolution, and also you're able to achieve a much higher degree of magnification. So the light microscope only allows us to see the relatively larger structures within a cell. However, the electron microscope allows us to see much smaller or finer structures within a cell. So let's compare the advantages and disadvantages of a light or optical microscope as compared to an electron microscope. And again, it's useful for you to be aware of these distinctions. So this table here illustrates some of the comparative advantages and disadvantages of optical versus electron microscopes. Now, as we'll see, there are two forms, general forms of electron microscopy. There is transmission electron microscopy and there is scanning electron microscopy. Now, we're going to get into those in a bit more detail shortly, but please also be aware that there are other more advanced forms of microscopy as well, which we're not going to get into in this webinar, because they go significantly beyond the content of A-level biology specifications. So you can talk about things like atomic force microscopy as well, but we're not going to go into that here. Okay, so let's have a look at electron microscopes and how they work in general. So electron micrographs will show specimens that have been much more highly magnified than you find from optical micrographs. Now, in order to gain a sense of proportion and size from such images, it's necessary to be able to interpret the magnification data and scale lines which are drawn on the image. Now, again, magnification is a value that tells us how much larger an image is when compared to the original object. So this is the equation which allows us to calculate and define the term magnification. Now, whenever you're using this equation to calculate magnification, it's important to remember that the size of the image and the size of the object must be substituted into this equation in the same units. So, for example, you can't put the size of the image in millimeters here and put the size of the object in micrometers here, because you would then get an incorrect value for the magnification. They must both be placed in as millimeters or both be placed in as micrometers. So, for example, if an image measures 30 millimeters and the specimen or the object measures 3 millimeters, PowerPoint has done its typical thing of converting my mu symbol into an M for millimeters here. So, sorry about that there. So, what this should show here is 30 micrometers here. So, if an image measures 30 or rather, if the object measures 3 micrometers here and the image measures 30 millimeters here, then we need to convert the image value into micrometers before we put it into this equation. So again, using those conversions that we saw before, 30 millimeters is equivalent to 30,000 micrometers, because again, 1 millimeter is 1,000th of a meter, whereas 1 micrometer is 1/1,000,000th of a meter. So we must place these in the same units into this equation. So in this case, the magnification is 30,000 micrometers for the size of the image divided by 3 micrometers for the size of the object, and this gives us a magnifying power of times 10,000. So the actual size of an object can be calculated using the same formula but rearranged. So if we rearrange this equation to make the size of the object the subject of this equation, we get this. So for example, if an image is measured and found to have a length of 6 millimeters, in other words 6,000 micrometers, and the magnification being used is times 40,000, what would be the size of the object? Now again, this is an example that appears in your student study and revision workbooks for this webinar. So hopefully those of you who have not uploaded your work for marking to your private coaching areas have at least attempted this. If you haven't, or if you're watching this as a YouTube recording, please do pause the video and attempt this calculation for yourself. Assuming that you've done that, let's have a look at how this is done. So we're trying to calculate the actual size of the object. So again, we would simply substitute these values into the equation, we evaluate, and we get 0.15 micrometers. So I said that there are two different kinds of electron microscopy. There are two different manners in which electron microscopes work. There is transmission electron microscopy and there is scanning electron microscopy. So let's have a look at those two types of electron microscopes and how they work. So let's start with the transmission electron microscope. Now, the transmission electron microscope, or TEM, this is a microscope that uses a beam of electrons rather than light to form an image, and this is true also of the scanning electron microscope. Now, this was developed in the 1930s in order to improve the resolution of images produced by microscopes. So the short wavelength of the electron beam allows for a higher resolution than optical microscopy and magnifications of up to 170 times greater than those obtained by even the best light microscopes. So the image in a transmission electron micrograph can be magnified to up to 500,000 times and allows for much finer detail and also a greater depth of field to be observed in the image. Now, complex procedures are required to prepare the specimen for observation with the transmission electron microscope. So we start by taking very, very thin sections of material, and these are then stained in solutions of heavy metals to produce contrast in the observed image. So how does the transmission electron microscope work? So we start with a stream of electrons being produced by this cathode here. And the cathode is essentially a very thin piece of tungsten wire through which an electrical current is being drawn, and this is drawn to a point. This produces a cloud of electrons which are being emitted by the tungsten wire. Those electrons are then accelerated towards a positive electrode. So the cathode is negatively charged, but here we have a high voltage positive electrode, which is the anode, and the electrons are attracted towards the anode, and then the anode then focuses them and produces a stream of electrons through this gap, which then passes into this chamber. Now, this entire internal chamber of the electron microscope is evacuated to produce a hard vacuum. Now the reason for that is that if there was any gas particles of air or any other gas present inside this compartment, this would cause the electrons to be scattered, and we don't want that because we want the electrons to pass as a coherent beam through the specimen stage here. So the stream of electrons is then channeled through a series of electromagnets. Now these electromagnets produce a field which functions in the similar way to the lenses found in an optical microscope. So rather than using material glass lenses, we are using electromagnetic lenses to focus that beam of electrons. So we start with the condenser lens, and this condenses the electron beam into a very fine stream which passes through the specimen stage, which is held here. Now this is a transmission electron microscope in the sense that the electron beam passes through the very thin specimen. So in that sense, the electron beam is transmitted through the specimen. So the beam strikes the specimen and parts of it go through the specimen, whereas parts of it are absorbed or scattered by the thicker portions of the specimen. Now the transmitted portion is then focused by this electromagnetic lens here, which is called the objective lens. Now again, this is analogous to the objective lens found in an optical microscope, in the sense that in both cases the objective lens is responsible for focusing and magnifying the image. So in this case, the transmitted portion of the electron beam, the part of the electron beam that passes through the specimen, is focused by altering the intensity of the electromagnetic field produced by this electromagnet, the objective lens. Now the projector lens is another electromagnetic lens which is responsible for focusing the image produced by the transmitted and focused portion of the electron beam. So this is responsible for passing that electron beam, which has passed through the specimen and has been focused by the objective lens, onto a phosphorescent stage where the electrons strike produces an image which can then be viewed by the observer or captured as a photograph. So this is essentially how the transmission electron micrograph works. Now again, we're using electromagnets here to produce electromagnetic fields which can then alter the shape of this electron beam for the purposes of focusing and projecting the image onto the phosphorescent stage where it can be viewed as an image by the observer. So again, the whole column, the whole internal compartment here is maintained under a hard vacuum because the electrons do not have sufficient energy to pass through a gas and would be scattered by any gas or air molecules here, and that would in turn blur the image. So let's have a look at some transmission electron photomicrographs. So this electron micrograph has been obtained by, these electron micrographs have been obtained using a transmission electron microscope. You can see the scale bar here. This is an animal cell, and you can clearly see the fine detail of the ultrastructure here, and also you can see in this plant cell the fine detail, including the chromatin present in the nucleus, and also the internal ultrastructure of some of these organelles as well that you see around here. So this is a transmission electron micrograph of a fibroblast cell, and this shows the detail of the rough endoplasmic reticulum, and you can even see the individual ribosomes studding the surface of the rough endoplasmic reticulum here. You can also see the cavities of the reticulum. Again, this is the electron micrograph of a chloroplast obtained under a transmission electron microscope. You can see the individual thylakoid membranes making up the grana. You can see the ribosomes here. You can see the starch grains. You can see the lipid droplets. And you can even see the lamellae between adjacent grana. So let's have a look at the second type of electron microscope here, which is called the scanning electron microscope. So this is another application of the same principles that you saw in the transmission electron micrograph, but it's being used in a different way here. So This was developed in the late 1950s and early 1960s. Now, the scanning electron microscope is designed for the study of surfaces of solid objects, such as, for example, pollen grains and insects. Now, in contrast to the transmission electron microscope, which transmits the electron beam through the specimen to show its internal structure, the scanning electron microscope does something different. Instead, it scans the surface of a specimen to reveal the detail on that surface. In this case, the electron beam is not transferred through the specimen, it is bounced off the surface of the specimen and detected to produce a very detailed image of that surface. So the electrons don't pass through the specimen in this case, they are because the thickness of the specimen, and because also because the specimen in this case is coated with a layer of a heavy metal such as gold. So in this case, the electron beam is reflected off the specimen's surface as it strikes the specimen, and special detectors which will collect those scattered electrons, and those will then create electrical signals in those detectors, which are then passed to a computer which assembles those signals into an image which can then be viewed by the operator. And you can see such a scanning electron microscope being used here. Now, one of the advantages for viewing surfaces using a scanning electron microscope is that it can produce very three-dimensional looking images of the specimen surfaces. So you can see here one such image of an ant, and you can even see the fine hairs on the surface of the exoskeleton of the ant, and this shows the scale bar down here. So the three-dimensional structure of red and white blood cells can also be seen here, for example, and again you can see the scale bar here. Here we can see a leukocyte, here we can see a red blood cell. Now, the images produced by both scanning electron micrographs and transmission electron micrographs are monochromatic. They are black and white because, again, we're not using visible light to produce these images. We're using electrons. Now, electrons have a shorter wavelength. Again, if you're modeling them as waves rather than particles, electrons have a much shorter wavelength than the wavelengths of electromagnetic waves such as visible light, and so colour has no real meaning in this sense. However, the computers which are connected to scanning and transmission electron microscopes can produce artificially coloured images, which are coloured in order to highlight specific objects in the image from the background. So again, one such image is shown here with false colour. And here's another image showing a scanning electron micrograph of the surface of a pollen grain, and the pollen grain has been coloured using the computer to distinguish it from the surrounding structures. Now, there are several of these worked examples which you can find in your student workbooks which accompany this revision webinar for those of you who book tickets in advance. I'm not going to go into these in too much depth because I see that we're already reaching the 3-hour point of this webinar. So again, I'm going to skip past these in order to save a bit of time here, but by all means, those of you watching via YouTube, please do pause and attempt these examples for yourself, and follow along at your own pace by either slowing down the rate of play or pausing. So let's compare the two types of microscopes, or the three types of microscopes that we've looked at, and this diagram compares the three types here. So you can see here the light microscope uses a source of visible light, whereas the electron microscopes rather use a cathode gun, which is a source of electrons. We see here that we have a glass condenser lens which refracts and focuses the light onto the specimen stage here in the case of the optical microscope. In the case of both the scanning and transmission electron microscopes, we're not using glass lenses here, we're instead using toroidal electromagnets which produce a varying electromagnetic field which can be used to focus the stream of electrons either for transmission through the specimen in the case of the transmission electron microscope, or in the case of the scanning electron microscope to focus the beam so that it can be bounced off the image. Here we can see the differences in the way the specimen is oriented in these three different kinds of microscopes. Here we can see that all of these have objective lenses, But in the case of the optical microscope, the objective lens is a glass convex lens, whereas in the case of electron microscopes, again, we are using toroidal electromagnetic lenses. We have a projector lens in the case of the transmission electron microscope as compared to the eyepiece lens in the case of the optical microscope. In the case of the scanning electron microscope, the way that the beam is played over the surface of the specimen in order to create the image is by means of a beam deflector, which makes use of positively and negatively charged plates to bend the beam so that it's passed over the surface of the specimen. Now, in terms of the viewing of the image, the light microscope makes use of direct image viewing via the eyepiece lens. The transmission electron microscope, the image is produced by focusing the beam of electrons that is passed through the image or through the specimen onto a phosphorescent stage, which then produces an image which can be viewed directly by the operator. But in the case of the scanning electron microscope, once that beam has played and been bounced off the surface of the specimen, the scattered electrons are detected by numerous detectors, which then feed electrical impulses into a computer, which assembles those electrical impulses into a three-dimensional image, which can be viewed on the screen connected to the computer. Okay, everyone, it seems like we've reached the end of the 3 hours for this webinar. Again, in keeping with the format of the previous webinars, I will leave it up to the student attendees in the private Zoom webinar room. If you could all just let me know in chat whether you prefer me to continue. If so, I will continue and go into cell fractionation, ultracentrifugation, and then we'll cover the topic of transport across cell membranes, and then we'll finish off with that, because again, we're not going into cell recognition and the immune system in this webinar because there's already a video there on my website for you. However, if you'd prefer, then I can shut off the YouTube live stream, go into the Q&A session. I see that there are a number of questions that you guys have asked for me to address in the Q&A at the end of this webinar, and what I can then do is upload an additional video to your private coaching areas to cover cell fractionation and ultracentrifugation. Excuse me, losing my voice. And again, so please do express your preference. If you'd prefer me to continue, I see some of you are asking how much longer will it take. Well, probably another hour or so, I would imagine, if not an hour and a bit, just to cover the process of cell fractionation, ultracentrifugation, and also transport across cell membranes. I'd say that that probably is going to take us an additional hour to an hour and a half, but also bear in mind that you all have access to those full lesson videos in this course module where those topics are taught in depth. And of course, if you have any questions, you can always type those in either the comments on those pages, or if you'd like to ask those questions of me privately, you will have your own private coaching areas for this course module, and you can ask those questions and I'll respond within 24 hours. So again, please do let me know what your preferences are. I see at least 6 of you have expressed your preferences here, so I'll just wait until everybody has voted on that. So would you prefer me to continue? Again, it'll probably add an additional hour to an hour and a half to this webinar, and then we'll go into the Q&A session. Or if you prefer, I can stop now and upload an additional video to your private coaching areas, and we can get into the Q&A here. And again, those of you watching via YouTube, the Q&A will not appear on the YouTube video. So okay, it seems like most of you have opted for me to stop here and go into the Q&A session. Okay, so I will do that. So to those of you watching via YouTube, I hope that was useful to you. I hope that, and again, if you do have any questions and you're not enrolled on this course module and you're not a enrolled student on my website, by all means, please do type your questions on the YouTube channel on this video's page when I upload it, and I'll be happy to answer your questions there.